Hello and welcome to Sleepy Science. Tonight, you are about to discover how much of the ordinary world has been hiding in plain sight. Your body is producing invisible light. Ancient viruses are still written into your DNA. Storms above Earth can create antimatter, and somewhere in the darkness of space, metal may be falling from alien skies like rain. Across 100 strange but completely real scientific facts, we will travel from the hidden machinery inside your cells to animals that freeze and awaken again, forests connected beneath the soil, oceans with underwater lakes, and stars so dense that a single teaspoon of their matter would outweigh mountains. Some of these discoveries may change how you see your own body. Others may make Earth feel like a far stranger planet than the one you thought you knew. Before we begin, tell me in the comments where you are listening from and what time it is there. And don't forget to like the video and subscribe for more calm, mind-opening science. Now, allow the noise of the day to grow distant. Settle into the darkness around you, and let us begin with the invisible light already leaving your body. Your body emits a faint light that human eyes cannot see. Even in complete darkness, your body is not entirely dark. At this very moment, an extremely faint stream of light is leaving your skin. Not as heat and not as a supernatural aura, but as individual photons produced by ordinary chemical activity inside your cells. Researchers have photographed this hidden glow using cameras sensitive enough to detect light roughly 1,000 times weaker than the threshold of human vision. The emission is connected to metabolism, especially reactions involving oxygen. As your cells convert nutrients into usable energy, highly reactive molecules are sometimes formed, and when certain molecules return from an energized state to a more stable one, a tiny amount of their energy can escape as light. This phenomenon is known as ultra-weak photon emission, and it occurs in many living organisms, including plants, animals, and humans. Your brightness is not constant throughout the day. In controlled observations, the glow followed a daily rhythm, generally becoming stronger in the afternoon, even though body temperature tended to peak at a different time. Different areas of the body also emitted different amounts, with parts of the face appearing brighter than some other regions. None of this means that people shine like fireflies, or that cameras can reveal emotions, personalities, or mystical energy fields. The light is far too weak for your eyes to detect, and it is produced by measurable biochemical processes rather than anything paranormal. Yet the reality is still quietly remarkable. The body you experience as solid and silent is filled with countless reactions that occasionally release fragments of visible energy into the surrounding darkness. You never notice them, just as you never hear the chemical conversations inside your cells. But sensitive instruments can reveal that life is not completely sealed beneath the skin. Every breath supplies oxygen to reactions that keep you alive, and some of those reactions end by releasing a photon that travels away from you. The light carries no portrait of who you are, but it does reveal something profound about being alive. Your body is not a motionless object occupying space. It is an active process, continually transforming matter, releasing energy, repairing damage, and leaving traces too delicate for your natural senses to perceive. You move through each night believing darkness begins at the surface of your skin, while in reality, your living cells are producing a glow that remains just beyond the edge of sight. Ancient viruses make up part of the human genome. Buried inside your chromosomes are the remains of infections that happened before modern humans existed. Roughly 8% of the human genome is composed of sequences associated with endogenous retroviruses, genetic fossils left behind when ancient viruses infected the reproductive cells of distant ancestors. A normal viral infection may affect one individual and then disappear, but an infection of an egg cell, sperm cell, or their ancestral cells can have a far longer future. If viral genetic material becomes inserted into that cell's DNA and the individual later produces offspring, the insertion can be inherited like any other genetic sequence. Across millions of years, These viral fragments passed from generation to generation until they became permanent residents of the human genome. Most no longer behave like active viruses. Mutations have damaged their instructions, pieces have been deleted, and cellular systems usually keep them switched off. They cannot simply assemble themselves into infectious particles and escape from your body. They are more like ruined machinery whose parts remain embedded in an ancient factory wall. But evolution does not always discard what it inherits. Sometimes it modifies old material and gives it an entirely new purpose. One of the most striking examples involves proteins called synchrotins, which originated from viral envelope genes. Viruses once used related proteins to fuse with host cells. Mammals later adapted that same cell-fusing ability to help form the placenta, where cells merge into a specialized layer supporting the developing embryo. A mechanism that once helped a virus enter an ancestor eventually became useful in reproduction. Other viral-derived sequences can influence when nearby genes are activated, and some participate in immune regulation. Although researchers are still working to understand when such activity is helpful, neutral, or associated with disease, it would therefore be misleading to say that you are partly an active virus. You are instead carrying a vast evolutionary archive filled with broken genetic records of encounters that your ancestors survived. Your genome is not a perfectly designed instruction manual written in a single moment. It is a layered document edited by mutation, natural selection, duplication, migration, and infection. Some passages are essential, some appear inactive, and others have been repurposed so completely that their violent origin is easy to forget. When you imagine inheritance, you may picture traits flowing neatly from parent to child, but your DNA also contains messages carried through deep time by organisms that once invaded living cells. The boundary between an organism and its history is therefore less clean than it seems. Part of what allows a human body to develop today may depend on molecular tools inherited from viruses that disappeared millions of years ago, leaving behind not an illness, but a permanent sentence in the biological story that eventually became you. Thunderstorms can produce antimatter above Earth. Thundercloud can briefly become one of the most powerful natural particle accelerators on Earth. Inside certain storms, enormous electrical fields develop as collisions between ice particles and water droplets separate positive and negative charge. Most of the time, you notice the result as lightning, a brilliant channel through which the atmosphere suddenly releases electrical tension. But some storms produce another event that cannot be seen from the ground, an extremely short burst of high-energy radiation known as a terrestrial gamma-ray flash. Within the intense electric field of the cloud, a fast-moving electron can be accelerated to enormous energy. As it collides with air molecules, it releases energetic gamma rays, and some of those gamma rays carry enough energy to interact with matter and create a matched pair of particles, an electron and a positron. The positron is the antimatter counterpart of the electron. It has the same mass but the opposite electrical charge. This does not mean that a glowing reservoir of antimatter forms inside every storm, nor does it mean thunderstorms pose an exotic annihilation threat to people below. The positrons are produced in tiny quantities, survive only briefly, and usually meet ordinary electrons. When matter and antimatter meet, both can disappear in an annihilation event that converts their mass into gamma radiation with a distinctive energy signature. Instruments aboard NASA's Fermi Gamma Ray Space Telescope detected this signature and showed that some thunderstorms can send beams containing positrons upward into near-Earth space. In several observations, the spacecraft was not even directly above the source storm. Charged particles had apparently traveled along the curves of Earth's magnetic field before reaching the detector, revealing that the storm's influence could extend far beyond the cloud itself. Scientists are still investigating exactly how thunderstorm electric fields become strong enough to initiate these events, and why some storms generate powerful gamma-ray flashes while apparently similar storms do not. What is clear is that antimatter is not confined to distant galaxies, advanced laboratories, or the imagined engines of science fiction. Under the right conditions, it can be created naturally in the turbulent atmosphere above an ordinary planet. The next time thunder rolls across a dark horizon, the spectacle you witness may be only the visible layer of the event. Hidden above the rain, electrons may be accelerating almost to the speed of light. Gamma rays may be passing through the cloud, and fleeting particles of antimatter may be following Earth's magnetic field into the sky. A storm that appears ancient and familiar from the ground can, for a fraction of a second, reveal the same high-energy physics that operates in some of the most extreme environments in the universe. One species of jellyfish can return to an earlier stage of life. A tiny jellyfish drifting through the sea can perform a biological reversal that no human body has ever achieved. When injured, weakened, or threatened by harsh conditions, Turritopsis dohoney may abandon its adult form and begin moving backward through its own life cycle. Most jellyfish develop from stationary structures called polyps into free-swimming adults known as medusae, and that transformation normally travels in only one direction. This species can disrupt the sequence. Its bell contracts, its tentacles disappear, and the animal settles into a cyst-like mass attached to a surface. Cells within this structure are reorganized, and some change from one specialized type into another through a process called transdifferentiation. Eventually, new polyps can emerge and produce another generation of medusae, allowing the organism to begin the adult phase again. Because this reversal can potentially occur more than once, the species is often called the immortal jellyfish, but that name requires caution. It is not indestructible. An individual jellyfish can still be eaten, infected, starved, or killed before completing the transformation. Its unusual ability, therefore, does not guarantee endless life. It offers repeated escape from the ordinary one-way path of aging under suitable conditions. Scientists are studying its genome and gene activity to understand how it repairs DNA, reorganizes tissues, controls cellular identity, and coordinates such a complete transformation without losing the structure of the organism. The discovery quietly challenges the idea that biological development must always resemble a road with no return. In most animals, maturity closes doors behind the body, making earlier forms inaccessible. In this jellyfish, some of those doors remain unlocked. You are not looking at a creature that simply refuses to die, but at one whose cells can reconsider what they have become, dismantle an adult body, and rebuild life from an earlier chapter. Wood frogs can freeze for winter and return to life. Beneath a quiet layer of fallen leaves, a wood frog can spend part of winter with no heartbeat, no breathing, and much of the water in its body turn to ice. This sounds like death. Yet the frog has entered a controlled state of extreme biological suspension rather than truly dying. As temperatures fall and ice begins forming around its body, the freezing spreads into spaces outside its cells. Water is drawn outward, reducing the chance that sharp ice crystals will grow inside the cells and tear their delicate structures apart. At the same time, the frog rapidly releases large amounts of glucose from stored glycogen in its liver, while substances including urea also contribute to protection. These natural cryoprotectants help stabilize cells, limit dangerous water loss, and reduce damage as circulation stops, and organs are deprived of oxygen. A large proportion of the frog's total body water may freeze, its limbs become rigid, its eyes appear lifeless, and ordinary signs of activity disappear. Yet its cells remain chemically protected and prepared for the return of warmth. When spring temperatures rise, thawing must occur carefully. The heart resumes beating, blood begins moving again, breathing returns, and damaged molecules and tissues require repair as normal metabolism restarts. This recovery is not effortless, and wood frogs cannot survive every temperature, every freezing duration, or every pattern of thawing. Populations from colder regions often possess stronger seasonal adaptations than those living farther south, showing that even this remarkable ability has limits shaped by local environments. What you witness is therefore not resurrection in the supernatural sense, but an extraordinary solution to a physical problem that would kill most vertebrates. The frog survives not by preventing winter from entering its body, but by controlling where the ice forms and preparing its cells for stillness. For months, life becomes almost invisible, held not in movement or breath, but in preserved structure, waiting beneath the snow for the conditions that will allow the body to continue. Octopus arms can process information without waiting for the brain. Touch an octopus arm with something unfamiliar, and the limb does not need to report every detail to a central brain before beginning to investigate. Running through each arm is an extensive network of nerve cords, local ganglia, sensory neurons, and motor circuits capable of processing touch, chemical information, sucker activity, and parts of movement close to where those signals are received. This distributed arrangement helps solve an enormous control problem. Unlike your limbs, which are shaped by bones and joints, an octopus arm is soft and can bend, twist, shorten, lengthen, stiffen, and form temporary curves almost anywhere along its body. Directing every muscle fiber from the brain in real time would require a staggering amount of computation. Instead, the central brain can issue broader goals, such as reaching toward an object, while neural circuits within the arm help organize the changing pattern of bends, muscle contractions, and sucker responses needed to carry out the action. Experiments have shown that basic extension patterns can be produced by circuitry embedded in the arm, and recent anatomical studies reveal repeating processing centers along the axial nerve cord, as well as specialized ganglia associated with individual suckers. Yet describing each arm as an independent brain would be misleading. The arms exchange information with the central brain and with one another, and coordinated behaviors such as hunting, crawling, learning, and directing an object toward the mouth depend on cooperation across the whole nervous system. The arm possesses local autonomy, not a separate personality or proven consciousness. Still, its design forces you to reconsider where an animal's intelligence must reside. In humans, thought appears concentrated behind the eyes, making the body seem like a collection of tools controlled from above. In an octopus, sensing and control are spread through the animal, allowing its limbs to participate directly in decisions about contact and movement. The octopus does not merely have a brain that controls 8 arms. It has a nervous system whose intelligence is partly woven into the arms themselves. A natural nuclear reactor operated beneath Africa 2 billion years ago. Long before human beings learned how to control nuclear fission, a deposit of uranium beneath what is now Gabon quietly began doing it on its own. The evidence was discovered in 1972, when scientists examining uranium from the Oklo mines noticed that it contained slightly less uranium used for sustaining fission than natural ore should contain, as though some of the fuel had already been consumed. Around 2 billion years ago, the proportion of this uranium isotope was much higher than it is today, and geological processes had concentrated enough of it within layers of rock for a chain reaction to become possible. Groundwater then entered the deposit and slowed newly released neutrons, making them more likely to split additional uranium nuclei and continue the reaction. As the rock heated, some of the water would boil away, weakening or stopping the process. After the area cooled and water returned, fission could begin again. The natural reactor probably operated in repeated pulses over a very long period, producing heat and radioactive byproducts without machinery, engineers, or a constructed containment chamber. Remarkably, many of those products moved only limited distances through the surrounding geology, which is one reason Oklo has been studied as a natural example of how radioactive material may behave underground across immense spans of time. Nature did not build a power station in the human sense, and no electricity was generated, but the essential physical process was real. Uranium atoms split, neutrons were released, and a self-sustaining reaction occurred. Oklo reminds you that scientific laws did not begin when people discovered them. Beneath an ancient landscape with no cities, instruments, or observers, the planet had already assembled the right materials and conditions to perform an experiment that humanity would not understand for nearly 2 billion years. Ancient supernova dust has been found on Earth's ocean floor. Hidden inside slowly accumulated material on the ocean floor are rare iron atoms that appear to have arrived from an exploding star. Scientists identified a radioactive form called iron-60 in deep-sea sediments and in ferromanganese crusts, mineral layers that grow extremely slowly and preserve a timeline of material settling from the ocean above. This isotope is important because Earth does not naturally produce it today in enough quantity to explain the detected signal, and it decays over a few million years. If iron-60 is still present in geological layers only a few million years old, it must have been created relatively recently on a cosmic scale, most likely inside massive stars and expelled when one or more of them exploded nearby. The debris did not strike Earth as a dramatic sheet of visible ash. Tiny dust grains traveled through interstellar space, entered the solar system, passed through the atmosphere, and became mixed into oceans before settling into sediments. In some samples, the iron was later incorporated into microscopic magnetic crystals produced by bacteria, meaning living organisms help preserve a chemical trace of a stellar death. Measurements indicate an extended period of deposition beginning roughly 2.5 to 3 million years ago, rather than one sudden moment, suggesting that supernova dust may have reached Earth gradually as the solar system moved through the scattered remains. Scientists are careful not to claim that this event caused a particular extinction or climate change, because the geological evidence does not establish such a direct connection. What it does establish is already extraordinary. The ocean floor is not only a record of Earth's history, it also acts as a quiet astronomical archive, collecting individual atoms from events that occurred far beyond the planet. A star disappeared somewhere in the nearby galaxy, and millions of years later, part of its remains became embedded in the mud beneath the Pacific, waiting for human instruments sensitive enough to recognize where it came from. The moon drags an invisible tail through space. The moon does not move through space as a perfectly sealed sphere. It continually loses atoms that form a vast, almost invisible tail extending far beyond its surface. The tail is made largely of neutral sodium released from lunar soil when micrometeorites strike, energetic particles from the solar wind hit the ground, or sunlight transfers enough energy to free atoms from the surface. Once sodium escapes into the moon's extremely thin exosphere, pressure from sunlight pushes it away from the sun, stretching the atoms into a stream hundreds of thousands of kilometers long. This does not mean the moon carries a bright tail like a familiar comet, and the stream does not simply trail behind its orbital motion like smoke behind a vehicle. Its direction is controlled mainly by the sun, and its density changes as impacts, solar activity, and lunar conditions vary. Around each new moon, Earth passes through this extended sodium stream. Earth's gravity then focuses some of the atoms into a narrow region, allowing specially designed cameras to detect a faint glow in the sky opposite the moon. Human eyes cannot see it because the emission is extraordinarily weak, but instruments tuned to sodium's characteristic light can map its shape and brightness. Long-term observations suggest that the tail is a permanent lunar feature, even though it becomes stronger or weaker from month to month. This hidden structure changes the way you might picture an airless world. The moon appears still and untouched when seen from Earth, yet its surface is constantly interacting with sunlight, meteoroids, and charged particles, releasing matter into interplanetary space. It is not surrounded by a substantial atmosphere, but it is not completely isolated either. Every month, without sound or visible motion, Earth passes through a trace of material shed by its companion, while the moon's sodium continues drifting outward as a delicate record of the forces acting upon its surface. Earth's outer atmosphere extends beyond the moon. The boundary of Earth is far less definite than the blue curve you see in photographs from space. Beyond the familiar layers where aircraft fly and weather forms, the atmosphere gradually fades into an enormous cloud of extremely sparse hydrogen called the geocorona. By examining ultraviolet observations collected by the ESA and NASA Solar and Heliospheric Observatory, researchers found traces of this hydrogen reaching about 630,000 kilometers from Earth, nearly twice the average distance of the Moon. This means the Moon travels within the most distant outer region of Earth's atmosphere. Although calling it air can create the wrong impression, the hydrogen atoms are separated by such enormous distances that this region would still feel like an almost perfect vacuum. There is no breathable gas, noticeable wind, or atmospheric pressure surrounding the Moon. Scientists detected the geocorona because hydrogen atoms scatter a particular kind of ultraviolet sunlight that human eyes cannot see. Solar radiation also affects its shape, compressing the cloud on the sun-facing side and stretching it farther into space on the opposite side. The discovery matters because this faint hydrogen can influence astronomical observations and may help scientists understand how planets slowly lose atmospheric material. It also changes the simple image of Earth as a sealed sphere wrapped in a thin shell. Our planet does not end abruptly at a fixed altitude. It becomes quieter, thinner, and more difficult to detect until individual atoms are drifting through a region usually described as outer space. The Moon may look completely separate from Earth, yet it spends its entire orbit moving through the last delicate traces of the planet below. Some sea slugs steal chloroplasts and use energy from sunlight. For certain sea slugs, eating algae is not only a way to obtain food, it is also a way to acquire temporary solar equipment. Sacoglossan sea slugs pierce algal cells and drink their contents. But instead of digesting everything, some species preserve the chloroplasts, the structures algae normally use to capture light and perform photosynthesis. The stolen chloroplasts are stored within cells lining the slug's digestive system, where they may continue functioning for weeks or even months. This remarkable process is called kleptoplasty, meaning the theft and retention of plastids. When light reaches the slug, the chloroplasts can manufacture energy-rich compounds that contribute to the animal's nutrition, survival, growth, or reproduction. Experiments have shown that photosynthesis can improve reproductive investment in some species. But the slugs are not truly plants and cannot usually survive forever on sunlight alone. Chloroplasts normally depend on many genes located inside the algal cell nucleus, which the slug does not steal, so their continued operation is especially puzzling. Genomic studies have found no convincing evidence that the slug simply transferred the necessary photosynthesis genes into its own chromosomes. Researchers instead suspect that unusually durable chloroplasts, protection from damaging light, and conditions inside the slug's cells allow the captured structures to remain useful for a limited time. Eventually, they lose function and must be replaced by feeding on more algae. The slug therefore does not create photosynthesis from nothing. It borrows working machinery built by another organism and keeps it alive inside an animal body. In doing so, it blurs a boundary you may have assumed was permanent, showing that evolution can produce a creature that hunts like an animal, while carrying fragments of a plant's relationship with sunlight beneath its skin. Certain fungi can control the behavior of living insects. Worker ant can leave the safety of its colony, climb vegetation, and lock its jaws onto a leaf at almost exactly the location where a fungus needs it to die. This disturbing sequence is associated with specialized fungi in the Ophiocordyceps group, parasites that have evolved close relationships with particular ant species. Infection begins when fungal spores attach to an ant and penetrate its outer body. The fungus then grows through the host, drawing nutrients from it and altering the animal's normal behavior. Instead of continuing its ordinary tasks, the infected ant may wander away from the nest, climb to a height with suitable temperature and humidity, and perform a powerful final bite that anchors its body in place. After the ant dies, the fungus develops a reproductive structure that emerges from the body and releases spores toward the forest floor, where other ants may encounter them. The result appears like deliberate puppetry, but scientists do not yet know every chemical and neurological step involved. Studies suggest that fungal cells can spread densely around muscles and other tissues, while leaving the brain itself physically uninvaded. Meaning control may involve secreted molecules, disrupted communication, altered muscle activity, and changes in the ant's internal rhythms, rather than a fungus simply replacing the brain. The manipulation is also highly specific. A fungus adapted to one ant species may kill another without producing the same precise behavior. This reveals a long evolutionary contest in which the parasite succeeds only when it can influence its host at the correct time and place. The ant is not consciously obeying an external commander, yet its final actions serve the reproductive needs of another organism. What looks like an individual decision can therefore be the visible outcome of an invisible biological struggle taking place throughout the body. Slime molds can solve mazes without having a brain. Place food at two exits of a maze. Release a slime mold inside, and something without a brain, nerves, or even a fixed body can gradually reveal an efficient route between them. The organism used in the famous experiment, Physarum polycephalum, spends part of its life as a large spreading cell containing many nuclei. It moves by extending a web of soft tubes through which its internal fluid flows rhythmically, allowing nutrients and chemical information to circulate across the entire structure. When researchers distributed the slime mold throughout a maze and placed food at two locations, it initially occupied many passages, exploring broadly rather than knowing the answer in advance. Over time, however, routes that transported material efficiently became thicker, while poorly performing branches narrowed and disappeared. The surviving network connected the food sources through a path close to the shortest available route. Calling this solving does not mean the organism studies the maze, imagines alternatives, or makes conscious plans. Its apparent decision emerges from local physical feedback. Useful tubes receive stronger flow and are reinforced, while inefficient ones are abandoned. This same process can help Physarum connect several food sources with networks that balance short travel distances against resilience, which is why researchers have used its behavior as inspiration for transport and computer models. What makes the organism so instructive is not that it secretly thinks like an animal, but that complex problem-solving can emerge without a central commander. You may be accustomed to locating intelligence inside a head, yet the slime mold shows how a living system can compare possibilities through movement, reshape itself according to success, and arrive at an effective answer without ever representing the problem in a mind. Bacteria count their population before acting together. A single bacterium may remain quiet while surrounded by empty space, then change its behavior dramatically once enough of its own kind have gathered nearby. It does not count neighboring cells one by one. Instead, many bacterial species use a chemical process known as quorum sensing. Individual cells continually release small signaling molecules into their surroundings, and at low population density those molecules diffuse away before becoming concentrated. As the community grows, the signals accumulate. When their concentration crosses a threshold, receptors inside or on the surfaces of the bacteria detect the change and activate particular sets of genes across much of the population. This allows thousands or millions of separate cells to begin a coordinated activity at roughly the same time. Depending on the species, quorum sensing can regulate light production, the construction of protective biofilms, the release of digestive enzymes, movement, competition with other microbes, or factors involved in infection. The classic light-producing bacteria associated with certain marine animals benefit from waiting because one glowing cell would achieve almost nothing, while a dense population switching on together can create visible illumination. Other bacteria use several signals, distinguish messages from different species, or interfere with the chemical conversations of competitors, so the system is far more complicated than a simple universal alarm. It is also important not to imagine bacteria holding intentions or conducting meetings. Each cell follows molecular rules, but those rules allow the population to behave like a coordinated group when acting alone would waste energy or fail. Quorum sensing reveals that cooperation does not require language, awareness, or leadership. Sometimes a community begins with nothing more than molecules accumulating in silence until every cell receives the same message. There are enough of us now. Plants release chemical warnings that nearby plants can detect. When a caterpillar begins chewing a leaf, the damage may alter the air around the plant before any neighboring leaf is touched. Wounded and herbivore-attacked plants can release changing mixtures of volatile organic compounds, small airborne molecules that travel away from the damaged tissue. Nearby plants exposed to some of these compounds may adjust gene activity, increase defensive chemicals, or enter a primed state in which they respond more rapidly and strongly if an attacker later reaches them. Researchers have observed this effect in controlled experiments involving crops, wild plants, and specific airborne compounds, and modern imaging has even revealed internal calcium responses, beginning after leaves perceive certain damage-related volatiles. These chemicals can serve several functions at once. Some discourage herbivores directly, while others attract parasitic wasps or predators that attack the insects feeding on the plant. Yet describing the process as plants consciously warning their friends would go beyond the evidence. Plants do not compose messages with deliberate meanings. Every neighboring species does not respond identically, and wind, distance, temperature, microbial activity, and the exact chemical blend can all change the result. A receiver may also benefit from detecting these molecules whether or not the damaged plant gains anything from releasing them. The safer conclusion is that plants can extract useful information from chemicals produced during danger. Rooted in place, they cannot escape an approaching insect, but they are not cut off from events occurring around them. They continuously sample light, touch, moisture, gravity, soil chemistry, and airborne molecules, turning those signals into changes within their tissues. What appears to you as a silent patch of vegetation may therefore contain an unseen exchange of information, not spoken in thoughts or words, but carried from leaf to leaf through the moving air. Tiny crystals inside your ears. Tell your brain which way is down. You can close your eyes, tilt your head, and still know which direction the ground lies because thousands of microscopic crystals are shifting inside your inner ears. These grains, called otoconia, are made largely of calcium carbonate and rest on a soft, jelly-like layer within two small structures known as the utricle and saccule. Beneath that layer are delicate sensory hair cells connected to nerves. When you lean forward, rise in an elevator, accelerate in a car, or simply stand under the pull of gravity, the relatively heavy crystals lag or slide slightly, bending the hair cells below them. That mechanical movement is converted into nerve signals, giving your brain information about head position and straight line motion. Your brain then combines these messages with vision, signals from muscles and joints, and information from the semicircular canals, which mainly detect rotation. Balance is therefore not a single sense, but a carefully negotiated agreement between several systems. When otoconia become dislodged and enter a semicircular canal, they can stimulate it at the wrong time, producing the spinning sensation of a common condition called benign paroxysmal positional vertigo. A movement as small as rolling over in bed may then make the room seem to turn. Most of the time, however, these crystals work so quietly that you never know they exist. Every step depends partly on tiny stones moving through fluid in darkness, reminding you that your feeling of being upright is not an obvious truth delivered directly by the world. It is a conclusion your brain continually builds from hidden motion deep inside your skull. Your bones produce electrical signals when they bend. Each time your foot meets the ground, your skeleton does more than carry the impact. The slight deformation of bone can generate tiny electrical changes within the tissue. Bone may feel rigid, but on a microscopic scale, it is a flexible composite made from mineral crystals, water, living cells, and an organized framework of collagen. When mechanical force bends or compresses this structure, electrical charge can develop partly because collagen has piezoelectric properties, meaning deformation can alter the distribution of charge within it. Movement of charged fluid through the microscopic pores of hydrated bone can produce additional electrical effects known as streaming potentials. These signals are extremely small and do not travel through your skeleton like the current in a household wire. Yet they form part of the physical environment sensed by bone cells. Osteocytes, which live embedded throughout the mineralized tissue, are especially important detectors of strain and fluid movement. They help communicate where bone is regularly loaded, where it is underused, and where microscopic damage may require attention. The body can then adjust its remodeling activity, strengthening regions that repeatedly bear force while allowing unnecessary material elsewhere to be removed. Scientists still debate exactly how much each electrical mechanism contributes to this response, so it would be too simple to claim that electricity alone tells bones how to grow. Mechanical strain, hormones, nutrients, cellular signals, age, and activity all work together. Even so, the electrical behavior is real and has inspired medical research into stimulation methods and piezoelectric materials designed to support bone repair. Your skeleton is therefore not passive scaffolding. With every walk, lift, and change of posture, pressure becomes motion, motion alters charge, and living tissue reads the consequences of how you move through the world. Your skeleton is continually destroyed and rebuilt. The bones supporting you today are not exactly the same tissue that carried you through earlier years, because your skeleton is always undergoing controlled demolition followed by reconstruction. This lifelong process is called bone remodeling, and it occurs in small working areas rather than replacing every bone at once. Specialized cells called osteoclasts attach to selected surfaces and dissolve old or damaged mineral while breaking down the underlying matrix. After they finish, osteoblasts arrive and produce a fresh collagen-rich framework called osteoid, which gradually hardens as minerals are deposited into it. Some osteoblasts become trapped within the material they created and mature into osteocytes, cells that remain connected through tiny channels and help monitor mechanical stress. Remodeling repairs microscopic cracks created by everyday movement, adapts bones to changing physical demands, and helps regulate the supply of calcium and phosphate needed elsewhere in the body. The balance is delicate. During growth, formation can exceed removal. Later in life, resorption may gradually become greater, especially when hormonal changes, inactivity, poor nutrition, illness, or certain medications disrupt the cycle. Osteoporosis develops when too much bone is lost or too little replacement is formed, weakening the internal architecture even while the outside may appear unchanged. This renewal also explains why exercise matters. Bones respond to regular loading, while prolonged weightlessness or immobility can encourage loss. Yet remodeling is not a complete reset that erases every trace of age, and not every part of the skeleton renews at the same speed. What it reveals is more subtle. The structure you think of as permanent survives through continuous change. Your skeleton remains strong not because it resists all destruction, but because healthy tissue knows when to remove what has become worn and when to quietly build again. 2 meters of DNA fit inside a single microscopic human cell. Inside one of your microscopic cells lies a molecule so long that, if carefully stretched out, it would measure roughly 2 meters from end to end. This is the DNA contained in the nucleus of most nucleated human cells. Yet the nucleus itself is only a few millionths of a meter across. The molecule fits because it is not pushed inside as a tangled thread. DNA first wraps around clusters of proteins called histones, forming repeating units that resemble beads along a string. These units fold into chromatin, which bends, loops, and attaches to organizing proteins that keep distant regions arranged within particular areas of the nucleus. The packing must solve 2 opposing problems at once. DNA has to remain compact enough to fit, but accessible enough for enzymes to read genes, copy the genome before cell division, and repair damage. Some regions are kept relatively open when their instructions are needed, while others are folded more tightly and remain mostly inactive. During cell division, the material becomes even more condensed, producing the familiar chromosome shapes seen in microscope images. This organization is not merely a storage trick. The way DNA is folded can influence which genes a cell uses, helping a liver cell behave differently from a neuron, even though both contain nearly the same genetic library. Small changes in these loops can bring regulatory regions closer to genes or move them farther away, altering biological activity without rewriting the genetic letters themselves. The astonishing part is therefore not only that 2 meters fit inside a cell, but that the cell can repeatedly locate the correct section among billions of DNA letters while keeping the rest protected. Within almost every nucleus, an immense molecular archive is being folded, opened, read, copied, and repaired without ever becoming visible to you. Your body is built from instructions that would be far too long to fit if life had not learned how to turn extraordinary length into ordered, usable form. The brain processes pain, but its own tissue cannot feel pain. The organ that creates every pain you have ever experienced can itself be touched without feeling pain in its own tissue. Your brain interprets injury by receiving electrical and chemical signals from nociceptors, specialized sensory endings located throughout much of the body, but the brain tissue itself contains no ordinary pain receptors. This is why surgeons can sometimes perform parts of brain operations while a patient is awake after the scalp and other sensitive tissues have been numbed. The surgeon may stimulate exposed areas of the brain while the patient speaks, moves a hand, names pictures, or describes sensations, allowing vital regions to be mapped and protected. Yet this does not mean that everything inside your skull is painless. The scalp, skull covering, membranes surrounding the brain, major blood vessels, and some cranial nerves can all generate pain signals. Headaches usually arise from these surrounding structures. Changes in blood vessels, inflammation, muscle tension, or the activity of pain processing networks, rather than from the brain tissue aching directly. Once signals arrive, several brain regions work together to produce the experience. Some help locate the injury, others judge its intensity, and others add fear, distress, memory, and attention. This explains why pain is never a simple measurement of tissue damage. The same injury can feel stronger when you are anxious or exhausted, and weaker when attention is absorbed elsewhere. While chronic pain can continue after the original wound has healed, because the nervous system itself has become unusually sensitive. Pain is real, but it is constructed as a protective interpretation rather than detected as a finished object. The brain sits silently inside the skull, unable to feel a blade touching its own surface, yet capable of turning a small signal from your fingertip into urgency, suffering, and immediate action. The place where pain becomes conscious is therefore one of the few places that cannot directly experience the injury it so powerfully describes. Some people experience no mental images in their minds. Ask 2 people to imagine a red apple, and one may see a vivid fruit in the darkness behind closed eyes, while the other sees nothing at all. The absence or extreme weakness of voluntary mental imagery is known as aphantasia, and it reveals that human thought does not take the same form inside every mind. A person with aphantasia understands what an apple looks like, can recognize one instantly, and may describe its color, shape, and texture from knowledge, yet cannot deliberately create a picture of it in the mind. This is not blindness, poor eyesight, low intelligence, or a failure of imagination in the broader sense. Many people with aphantasia are creative, remember visual facts, solve spatial problems, and work successfully in art, science, design, or writing. They may think through words, concepts, relationships, bodily sensations, or an abstract sense of space rather than through internal pictures. The condition exists on a spectrum. Some people report complete darkness, others experience faint or unstable images, while at the opposite extreme, some minds generate scenes with remarkable clarity. Researchers often study it using questionnaires alongside tasks involving memory, perception, and brain activity. But there is still no single test that captures every person's inner experience. Voluntary imagery can also differ from dreaming. Some people who cannot form a picture while awake still report visual dreams, suggesting that deliberate visualization and dream imagery rely on partly different processes. Aphantasia may affect autobiographical memory in some individuals because recalling personal events often depends on reconstructing scenes, yet its impact varies widely, and many people do not realize they have it until adulthood. They assume that phrases such as "picture this" are merely figures of speech, while others have been silently seeing entire landscapes. The discovery invites a strange but useful question. How many apparent differences in memory, creativity, and communication arise not from what people know, but from the invisible formats in which they know it? You may share the same world with someone, use the same words, and reach the same conclusion, while the private path taken through the mind is completely different. Some people experience little or no inner voice. Not every mind contains a narrator describing the day from somewhere behind the eyes. Some people report an almost continuous inner monologue, while others experience words only occasionally, and a smaller number say that silent verbal thought is largely absent from their everyday awareness. This does not mean their minds are empty or that they cannot reason, plan, read, remember, or understand language. Thought can take many forms. A person may grasp an idea as a wordless relationship, feel the direction of a decision before naming it, recall facts without hearing them spoken, or move through a sequence of actions using spatial awareness and emotion rather than sentences. Researchers studying inner experience have found enormous variation between individuals, and moment-by-moment sampling suggests that even people who possess a strong inner voice are not necessarily using it all the time. Inner speech can help with self-control, rehearsal, problem-solving, emotional regulation, and preparing conversations. But it is only one tool available to the mind. Measuring it is difficult because private experience cannot be observed directly, and people may interpret questions such as "do you hear your thoughts?" in very different ways. An inner voice is also not the same as an external sound or an auditory hallucination. Most people recognize it as self-generated thought, even when it has a clear tone or conversational structure. The deeper lesson is that identical behavior can emerge from very different inner processes. Two people may pause, solve the same problem, and give the same answer, yet one may have silently debated each step, while the other arrived through a quiet, non-verbal sense of understanding. Language allows minds to meet in the outside world, but it does not prove that thought itself is always made of language. Beneath the words "people share," consciousness may be organized in ways that remain almost impossible for one person to imagine from inside another. Some blind people navigate by listening to their own echoes. A sharp click of the tongue can briefly turn a hallway, doorway, parked car, or tree into an audible shape. Some blind people practice human echolocation by producing short mouth clicks and listening to the faint echoes that return from nearby surfaces. The brain compares the outgoing sound with its reflection, using tiny differences in timing, loudness, frequency, and the way sound reaches each ear to estimate where an object is, how far away it may be, and sometimes whether it is broad, narrow, hard, soft, moving, or still. Large nearby objects often return stronger and earlier echoes, while smaller or more distant surfaces require greater sensitivity and repeated clicking. Skilled echolocators can use this information while walking, cycling, finding openings, avoiding obstacles, or recognizing features of unfamiliar spaces. Though performance varies greatly and depends on training, background noise, object material, and the acoustics of the environment. Brain imaging studies have produced an especially striking result. In expert blind echolocators, echoes can activate areas at the back of the brain, usually associated with visual processing. This does not mean the person literally sees in the ordinary sense. It shows that the brain is more flexible than the names of its regions suggest, capable of using areas normally devoted to sight to interpret spatial information arriving through sound. Echolocation is also learnable to some degree by many sighted and blind beginners, although it should complement rather than automatically replace tools such as canes, guide dogs, and navigation training. What begins as a small sound made by the mouth becomes a question sent into the surrounding space, and the returning echo becomes an answer. The walls do not speak, yet the brain learns to hear their presence, revealing that a map of the world does not always have to enter through the eyes. A fake rubber hand can begin to feel like part of your body. Hide your real hand from view. Place a realistic rubber hand where your own hand might naturally rest, and the brain can sometimes begin rewriting the border of the body within minutes. In the classic rubber hand illusion, an experimenter strokes the visible artificial hand while touching the participant's hidden real hand in the same places and at the same times. Your eyes report that the rubber fingers are being brushed, while your skin reports matching contact from approximately the same location. Faced with these synchronized signals, the brain searches for one explanation that can unite them and may conclude that the seen hand belongs to you. Participants can describe a vivid sense of ownership, and some begin estimating that their hidden hand lies closer to the rubber one than it really does. A shift known as proprioceptive drift. The illusion usually weakens when the strokes are poorly synchronized, when the model is positioned implausibly, or when its appearance is too inconsistent with a possible body part. This shows that the brain is not willing to adopt any object without limits. It tests visual, tactile, and positional evidence against an existing model of the body. Regions involved in combining touch, vision, and limb position help create the effect, turning body ownership into an ongoing calculation rather than a permanent label attached to flesh. The rubber hand does not truly connect to your nerves, and you do not lose knowledge that it is artificial. Instead, two levels of understanding briefly conflict. Intellectually, you know it is fake, while perceptually, part of the nervous system treats it as yours. This matters far beyond an entertaining laboratory trick. It helps researchers investigate phantom limbs, prosthetic embodiment, rehabilitation, virtual bodies, and the fragile mechanisms behind the feeling of self. The hand you call mine feels unquestionably personal, yet the experiment reveals that belonging is not sensed directly from the body. It is inferred from agreement among the senses, moment after moment, and under the right conditions, that agreement can extend the self into an object that was never alive. Water can boil and freeze at the same time. Water can appear to break its own rules when pressure is lowered until boiling no longer requires heat. Under ordinary conditions, you expect ice, liquid water, and vapor to belong to different temperature ranges, yet there is a precise combination of temperature and pressure called the triple point where all three phases can coexist in equilibrium. For pure water, this occurs just above 0 degrees Celsius and at a pressure far lower than the air pressure around you. In a carefully evacuated chamber, liquid water can begin boiling, even while it remains cold, because the reduced pressure allows molecules to escape from the surface much more easily. Those escaping molecules carry energy away, cooling the water that remains. If enough energy is lost, part of the liquid begins turning into ice, while another part continues becoming vapor, creating the startling sight of water boiling and freezing within the same experiment. This is not a contradiction, because boiling does not simply mean that a liquid is hot. It means its vapor can form bubbles under the surrounding pressure. Lower the pressure far enough and even cold water can boil. The triple point has been so reliable that specially prepared water cells were historically used to realize the Kelvin with extraordinary precision. Although the modern definition of the unit no longer depends on a physical sample, What you are watching is not water becoming confused, but matter responding faithfully to conditions your daily life rarely provides. Ice, liquid, and vapor are not fixed identities locked behind separate doors. They are arrangements of the same molecules, and a small change in pressure can bring those arrangements together at one delicate boundary, where something can vanish into gas, while another part quietly becomes solid. Sound waves can hold small objects suspended in midair. An object can float without magnets, wings, or a visible support when sound is shaped strongly enough to push against gravity. Acoustic levitation usually begins with a speaker-like transducer producing intense sound toward a reflector or another transducer. When the outgoing waves repeatedly meet waves returning in the opposite direction, they form a standing wave pattern containing stable regions where the acoustic forces can trap a small object. A lightweight bead, droplet, grain, or tiny biological sample placed near one of these regions can remain suspended because differences in sound pressure create an upward force that balances its weight. The object is not resting on a solid surface, and the air is not simply blowing it upward like a miniature fan. It is being held within an organized pressure field produced by rapid vibrations, often at ultrasonic frequencies too high for human hearing. By changing the timing, strength, or position of the sound sources, researchers can move, rotate, combine, or separate levitated samples without touching them. This contactless handling is valuable when a material might be contaminated by a container, react with a surface, or need to be studied while freely suspended. Scientists have used related systems to manipulate liquid droplets, small glass spheres, and materials undergoing heating or coating. The method still has limits. Larger or heavier objects require much stronger forces. Unstable airflow can disturb the trap, and powerful sound fields are not harmless merely because they are inaudible. Yet the underlying lesson is quietly surprising. Sound is usually experienced as information arriving at your ears, but physically it is moving pressure within matter, carrying momentum as well as music and speech. Arrange those vibrations with enough precision, and something invisible can become a temporary platform, holding a small piece of the world in open air. Metal can melt from the warmth of your hand. The solid piece of metal placed in your palm may begin collapsing into a shining liquid before your skin feels especially warm. Gallium behaves this way because its melting point is only about 29.8 degrees Celsius, lower than the temperature of a warm human hand. At an ordinary cool room temperature, a sample can remain solid, but once it absorbs enough heat from your skin, its crystal structure begins to loosen and the rigid shape slowly gives way. The change can look almost impossible because daily experience trains you to associate molten metal with furnaces, glowing crucibles, and dangerous heat. Gallium reveals that metal describes a family of elements with widely different properties, rather than a promise that every member must remain solid under familiar conditions. Its unusually low melting point comes from the way gallium atoms arrange and bond in the solid state, while the liquid remains stable across an enormous temperature range before boiling. The metal can also stay liquid below its normal freezing point if no suitable crystal begins the solidification process, a condition known as supercooling. Once freezing finally starts, gallium expands rather than shrinking, another feature that distinguishes it from many common metals. It can wet glass and skin, leaving a grey stain, and it can penetrate and severely weaken aluminum, so the hand-melting demonstration should never be performed over aluminum objects or valuable surfaces. Gallium is not the only low-melting metal, but it is one of the most memorable because its transition occurs so close to body temperature. In your palm, no law of nature has been suspended and no extraordinary heat has appeared. A boundary has simply been crossed. The object that seemed permanently solid was always conditional, waiting for only a few degrees of warmth to reveal that hardness is not an identity, but a temporary arrangement of atoms. Fetal cells can remain inside a mother's body for decades after pregnancy. Pregnancy can leave behind something far more enduring than memory. A small population of cells from the developing fetus may remain inside the mother for decades after the pregnancy has ended. During pregnancy, the placenta is not an absolute wall. Cells move in both directions across it, so maternal cells can enter the fetus, while fetal cells enter the mother's circulation. Most fetal cells disappear after delivery, but a very small number can survive, settle in tissues, and sometimes reproduce, creating a condition called fetal microchimerism. Researchers have detected fetal origin cells years or even decades later in blood, bone marrow, skin, liver, and other organs. Some early studies identified cells carrying a Y chromosome in women who had previously carried male fetuses, because those cells were easier to distinguish from the mother's own cells. But fetal microchimerism is not limited to pregnancies involving boys. The word microchimerism refers to the presence of a tiny genetically distinct cell population within another person, not to a second body living inside the mother. These cells are rare, scattered, and biologically complicated. Some appear capable of taking on characteristics of the tissues where they are found, and experiments have suggested that they may gather near wounds or areas of injury. This has led scientists to investigate whether they sometimes participate in tissue repair. Other studies have found associations between microchimeric cells and autoimmune diseases or cancers, but an association does not prove that the cells cause the condition. In some situations, they might contribute to inflammation. In others, they might respond to existing damage, and in many cases, they may remain harmless passengers. Researchers are still trying to understand how the immune system tolerates cells that are partly foreign, and why some survive while most are removed. The phenomenon, therefore, should not be turned into a sentimental claim that every fetal cell permanently protects the mother, nor into a frightening claim that pregnancy leaves dangerous invaders behind. The honest reality is more subtle. Pregnancy can permanently change the cellular population of the body, leaving a faint biological record that may persist long after birth. A mother may continue carrying a few cells that began in another developing body, while a child may also retain maternal cells acquired before birth. What appeared to be a clean separation at delivery is, at the microscopic level, not always complete. For years afterward, two biological histories can remain quietly interwoven, reminding you that the boundaries of an individual body are sometimes less absolute than they seem. The human liver can regrow much of its lost tissue. Remove a large portion of a healthy liver, and the tissue left behind can begin expanding within hours, responding as though the organ has measured what is missing. This ability makes the liver unusual among human organs, but the word regeneration can easily create the wrong picture. A removed lobe does not usually grow back from the cut surface as a perfect copy of its former shape. Instead, the remaining lobes enlarge until enough liver mass and working capacity have been restored. Mature liver cells called hepatocytes, which normally spend much of their time performing metabolic work rather than dividing, can re-enter the cell cycle after an injury or surgical removal. Other cell types, blood vessels, immune signals, growth factors, and changes in blood flow help coordinate the response. The remaining tissue suddenly receives a larger share of the blood that once passed through the whole organ, and this altered mechanical and chemical environment helps trigger growth. Existing cells become larger, many begin dividing, and the internal vascular network adjusts as the remnant expands. Studies of living liver donors show that volume can recover rapidly during the first weeks and months, while liver function may improve on a somewhat different timetable. One clinical study found that after the loss of about 61% of the original volume, the remnant had already regained enough size for essential function within 10 days, even though complete volumetric and functional recovery took longer. Other donor studies have shown that liver volume may remain below its exact preoperative measurement after a year despite normal function, demonstrating that the body restores what it needs, rather than rebuilding an identical anatomical replica. This power also has strict limits. Regeneration depends on how much healthy tissue remains, whether its blood supply is intact, and whether disease, scarring, infection, age, medication, malnutrition, or other complications interfere with growth. A cirrhotic or severely damaged liver cannot be assumed to respond like a healthy donor liver, and removing too much can cause fatal liver failure before regeneration has time to help. The liver's resilience is therefore not immortality and should never be interpreted as permission to damage it. Its achievement is quieter and more precise. It senses a dangerous loss of working capacity and asks ordinary adult cells to temporarily behave as builders. The organ survives not by preserving every piece of itself, but by allowing the portion that remains to change its priorities, divide, enlarge, and gradually carry the burden of the whole. Sharks existed on Earth before trees. There was a time when shark-like animals were already moving through ancient seas, while no true forest had ever risen on land. The famous claim that sharks are older than trees is broadly correct, although it needs one important distinction. The earliest evidence comes from members or possible members of the ancient cartilaginous fish lineage that eventually produced sharks, rays, and chimeras, not from familiar modern species such as great whites or hammerheads. Tiny fossil scales with shark-like features have been found in rocks from the late Ordovician period, around 450 million years ago. Because scales alone cannot reveal an entire animal, scientists still debate exactly how closely some of these early forms were related to true sharks. More complete evidence appears later. Fossil teeth belonging to early cartilaginous fishes are known from roughly 418 million years ago, while one of the oldest articulated chondrichthyan bodies dates to about 394 million years ago. Clear fossil evidence of tree-sized plants and early forests appears around 390 million to 385 million years ago during the Devonian. Before forests transformed the continents, land vegetation was generally lower and simpler. Soils were less deeply bound by roots, and terrestrial ecosystems lacked the towering trunks and layered canopies that now seem inseparable from the idea of nature. Meanwhile, the oceans had already supported vertebrate predators for tens of millions of years. Early shark relatives did not look exactly like the sharks swimming today, and the lineage repeatedly changed as species appeared, diversified, and vanished. Their skeletons were largely made of cartilage, which fossilizes less readily than bone. So much of their early history must be reconstructed from teeth, scales, spines, and rare preserved bodies. The phrase "sharks survived unchanged for hundreds of millions of years" is therefore misleading. Evolution never stopped working on them. Different shark groups survived environmental upheavals because their bodies, senses, reproduction, diets, and habitats continued adapting. Not because one perfect design remained frozen in time. Still, the timeline is astonishing. When the ancestors of trees had not yet built deep forests, the evolutionary story leading toward sharks was already unfolding beneath the waves. Long before branches filled the air, shadows with cartilage skeletons were moving through seas on a planet whose continents would have looked almost unrecognizable to you. Greenland sharks have been estimated to live for more than 4 centuries. Somewhere beneath the cold darkness of the North Atlantic, a Greenland shark may be carrying memories in its body from a century before electricity entered ordinary homes. These enormous, slow-moving animals inhabit deep Arctic and subarctic waters. Yet their age remained difficult to measure because sharks do not possess the layered ear bones commonly used to date many bony fish. The breakthrough came from an unusual part of the eye. Proteins at the center of a Greenland shark's lens form before birth and remain largely unchanged throughout life, preserving a chemical record from the period when the animal began developing. Researchers measured radiocarbon within the eye lenses of 28 female sharks and compared the results with historical changes in atmospheric carbon, including the distinctive increase caused by nuclear testing during the 20th century. Their study concluded that the species lives for at least about 272 years, while the largest shark, measuring just over 5 meters, received a midpoint age estimate of approximately 392 years. The uncertainty was very large, extending roughly 120 years in either direction, so scientists did not prove that this individual was exactly 392, or definitely older than 4 centuries. The finding instead established a credible possibility that Greenland sharks can survive for around 400 years, or perhaps longer, making them the longest-lived vertebrates currently known. The same research suggested that females may not reach sexual maturity until roughly a century and a half into life. Such delayed reproduction makes the species especially vulnerable, because a population that takes generations to replace mature adults cannot recover quickly from accidental capture or heavy fishing pressure. Researchers suspect that cold water, slow growth, and a low rate of energy use contribute to this longevity, but no single mechanism has yet explained how the animals maintain their tissues for so long. Their lives unfold at a pace almost incompatible with human intuition. A Greenland shark may spend more than a human lifetime simply approaching adulthood, then continue moving through darkness as nations rise, borders change, and several generations disappear above the water. Its age is still an estimate rather than a known birthday, but that uncertainty makes the animal no less remarkable. It asks you to consider how different time becomes when growth is measured not in years or decades, but in centuries, and when a single living body can quietly connect the modern ocean to a world its first human observers would no longer recognize. Dolphins can sleep with one half of their brain at a time. For a dolphin, falling completely unconscious in open water could mean failing to breathe, losing contact with its group, or becoming vulnerable before it wakes. Dolphins solve this problem through unihemispheric slow-wave sleep, a state in which one cerebral hemisphere displays the slow electrical activity associated with deep non-rapid eye movement sleep, while the other maintains a more wake-like pattern. The two sides alternate, allowing each hemisphere to receive rest without shutting down the entire animal at once. During these periods, the eye connected mainly to the more alert hemisphere is often open, while the opposite eye may close. This arrangement helps the dolphin continue monitoring its surroundings, controlling its position, and returning to the surface when necessary. Unlike human breathing, which continues automatically during ordinary sleep, a dolphin's breathing is closely coordinated with conscious surfacing, so remaining partly responsive is essential. The animal may swim slowly beside a companion, rest near the surface, or maintain gentle movement while half of the brain enters slow-wave sleep. This does not mean one side contains an awake personality while the other dreams independently, nor does it mean dolphins never require genuine neurological recovery. Experiments show that sleep pressure can build within an individual hemisphere, and that depriving one side of slow-wave sleep leads that side to compensate later, suggesting that the two hemispheres can regulate their need for rest partly independently. Rapid eye movement sleep, the state strongly associated with vivid dreaming in humans, appears extremely limited or unusual in cetaceans, possibly because the muscle relaxation accompanying it would create serious problems in water. Unihemispheric sleep also allows mothers and newborn calves to remain active and close together during demanding early periods, although their sleep behavior changes as the calf develops. What seems like an extraordinary mental trick is therefore a practical answer to the conditions of marine life. Evolution did not remove the dolphin's need for sleep. It reorganized sleep so that rest could occur without surrendering breathing, movement, and vigilance all at once. As one half of the brain slows, the other keeps a narrow doorway open to the ocean, and then the roles reverse. The dolphin continues traveling through water, while sleep passes quietly from one hemisphere to the other, showing that even a condition you experience as a total disappearance from the waking world can be divided, shared, and reshaped by the needs of a very different body. Crows can remember individual human faces for years. A face you forget after a brief encounter may remain important to a crow long after you have disappeared from its daily life. Researchers demonstrated this by wearing distinctive human masks while capturing and briefly handling wild American crows, creating an association between one particular face and a threatening experience. After the birds were released, people wearing the dangerous mask walked through the same areas at later dates, while other masks served as neutral comparisons. The crows did not simply react to every unfamiliar person. They were far more likely to scold, gather, follow, or dive toward the face connected with capture, and this response persisted for years. Because the mask preserved the same facial structure, while clothing and individual wearers could change, the experiments showed that the birds were identifying the face, rather than merely recognizing one researcher's body, walk, or smell. The knowledge also spread. Crows that had never been captured began responding after observing the alarm calls and defensive behavior of experienced birds, allowing information about a dangerous person to move through the local population. Younger crows could acquire the response socially as well, which helps explain why the number of birds reacting to the mask sometimes increased rather than fading as the original experience became more distant. Brain imaging studies later found different patterns of activity when crows viewed faces associated with threat and faces linked to caretaking, involving regions used for visual discrimination, memory, attention, and emotional evaluation. It is tempting to say that crows hold grudges, but that phrase gives them a specifically human motive that the experiments cannot prove. A safer explanation is that they possess durable recognition memory and use it to guide future decisions about risk. For a long-lived, social bird living around unpredictable humans, remembering exactly which person once captured or threatened a group member can be more useful than treating every human as equally dangerous. The ability also demonstrates that a small animal brain does not need to resemble yours to perform a socially sophisticated task. A crow can extract the stable identity hidden beneath changing clothes and movement, attach emotional importance to that identity, preserve the association, and pass the warning to others. When it watches you from a rooftop, you may see only another dark bird among many. It may be seeing an individual, placing your face within a history you did not realize it had kept. Elephants can detect distant vibrations through their feet. Long before an elephant appears on the horizon, another elephant may already know that it is coming because the ground has begun to speak beneath its feet. Elephant rumbles contain extremely low frequencies, and part of the energy from these calls can pass into the soil as seismic waves. Heavy footsteps and deliberate stamping can create ground vibrations as well. Depending on the type of soil, background noise, weather, and the strength of the original signal, these vibrations may travel much farther than an ordinary footstep would suggest. An elephant standing at a distance does not interpret them as a human interprets words through the ears alone. Anatomical studies have found large numbers of vibration-sensitive structures called Pacinian corpuscles in the padded feet of Asian elephants, particularly in regions that would be pressed against the ground when the animal shifts its weight forward or backward. These receptors are built to respond to rapid mechanical changes, making the feet plausible entry points for seismic information. Vibrations may also travel upward through the limbs and skeleton toward an inner ear that is well suited to low-frequency sound, so researchers think touch-like sensing and bone conduction may work together, rather than acting as completely separate channels. Elephants have been observed pausing, leaning, lifting a foot, or changing their behavior when exposed to relevant ground vibrations, although scientists are still investigating exactly what details they can extract under natural conditions. They may distinguish the movement of a herd, the approach of a threat, or the seismic component of social calls, but it would be an exaggeration to claim that every distant vibration carries a perfectly decoded message. The ground is noisy, wind moves trees, vehicles shake roads, rain strikes soil, and many animals generate footsteps. Useful sensing therefore depends on recognizing patterns within that confusion. What makes the ability remarkable is not simply that an elephant feels shaking. You can feel a truck pass nearby too. The elephant's body appears adapted to notice faint, low-frequency structures, that may remain meaningful across distances, where visual contact is impossible and airborne sound is weakened. Its feet are not only supports for several tons of weight, they are broad sensory surfaces pressed against a landscape full of hidden motion. As you watch an elephant standing almost perfectly still, it may not be resting or staring into emptiness. It may be attending to information traveling through the earth, listening with the part of its body you would least expect to hear. Reindeer eyes change color between summer and winter. When winter settles over the Arctic, the change does not stop at a reindeer's coat. Even the reflective surface deep inside its eyes shifts from gold toward a dark, intense blue. The colored structure is not the iris that surrounds the pupil, but the tapetum lucidum, a mirror-like layer behind the retina that sends incoming light back through the light-sensitive cells, giving those cells another opportunity to detect it. Many nocturnal animals possess a tapetum, which is why their eyes can appear to shine when illuminated at night. But reindeer perform an unusual seasonal adjustment. During the bright Arctic summer, when daylight may continue almost without interruption, the tapetum reflects a golden or turquoise color. In winter, after prolonged darkness keeps the pupils widely dilated for long periods, pressure inside the eye increases. Researchers found that this pressure appears to compress the organized collagen fibers within the tapetum, changing the spacing that determines which wavelengths are reflected. The result is a deep blue reflection. This altered structure sends less light directly back out of the eye and scatters more of it within the retina, increasing the chance that the scarce available light will reach photoreceptors. The winter eye therefore becomes more sensitive, helping the animal navigate, find food, recognize movement, and avoid danger beneath dim twilight, moonlight, and light reflected from snow. Greater sensitivity is not a free improvement, however. Scattering light more widely may reduce the sharpness of the image, creating a trade-off between detecting faint signals and preserving fine detail. In summer, when light is abundant, the eye can favor a clearer optical arrangement. In winter, gathering as much information as possible becomes more important than perfect precision. The transformation is gradual and physical, not a rapid color change the animal consciously controls. And it does not mean the entire visible eye becomes bright blue to anyone looking at the reindeer's face. The change is most obvious when light is reflected from the rear of the eye under examination. Still, it reveals how deeply the Arctic year enters the animal's biology. A reindeer does not merely endure months of brightness followed by months of darkness with the same sensory equipment. Its eye remodels its internal mirror to match the season, turning vision itself into something flexible. The world does not always become easier to see because more light appears. Sometimes the observer must quietly change the way light is allowed to move inside the body. Octopuses can rewrite RNA instructions inside their nervous systems. An octopus does not always follow a genetic instruction exactly as it was copied from its DNA. Before some instructions become proteins, the message can be altered along the way. DNA remains the long-term archive inside the cell, while messenger RNA is a temporary working copy carrying selected information toward the machinery that builds proteins. In a process known as RNA editing, enzymes can chemically change particular letters within that copy. One common form in animals converts a molecule called adenosine into inosine, which the protein-building machinery reads as though it were a different genetic letter. This can change one component of the resulting protein without permanently rewriting the DNA from which the message originated. Humans and many other animals also use RNA editing, so the basic mechanism is not unique to octopuses. What is unusual is its scale in behaviorally complex coleoid cephalopods, the group that includes octopuses, squid, and cuttlefish. Researchers comparing DNA and RNA found tens of thousands of editing sites, with especially extensive editing in nervous system tissues. Many of the affected instructions are connected to proteins involved in the electrical activity of neurons, communication between cells, and the structures that allow nerve cells to function. This gives the nervous system a way to produce different versions of certain proteins from the same underlying gene, expanding biological possibilities after the original message has already been copied. Later experiments showed that editing levels at many sites can shift when octopuses experience different water temperatures, and some of those changes alter proteins involved in neural transport and electrical signaling. The response is not instantaneous thought-powered self-reprogramming, and an octopus does not consciously choose which sentence of RNA to rewrite. Enzymes follow cellular conditions, sequence patterns, and regulatory signals. Nor does editing allow the animal to invent any protein it desires. It operates at particular sites within strict biochemical limits. The ability may also carry an evolutionary cost. For an editing site to keep functioning, the surrounding DNA sequence often needs to remain recognizable to the editing machinery, which can restrict how freely that region of the genome changes across generations. Octopuses may therefore gain flexibility in their temporary RNA messages while preserving unusually stable sections of DNA. This is not the same as rewriting inheritance, yet it challenges the simple picture of genes issuing fixed commands that cells merely obey. Between the permanent archive and the finished protein lies an editable draft, and in the octopus nervous system, that draft is revised with extraordinary frequency. The animal's complexity is not explained by RNA editing alone, but the process reveals a nervous system that can adjust some of its molecular tools without waiting for evolution to alter the original genetic text. Some turtles absorb oxygen through specialized tissues near their tails. A turtle resting beneath a river can draw oxygen from water through an opening most people associate only with waste. Certain freshwater turtles possess a pair of internal sacs called cloacal bursae, connected to the cloaca near the rear of the body. The walls of these bursae contain many folds, fine projections, and dense networks of blood vessels, creating a large surface across which dissolved oxygen can move from water into the bloodstream. Muscular movements draw fresh water into the cloacal chamber and push depleted water back out, repeatedly exposing the tissues to new oxygen. This is often jokingly described as breathing through the butt, but the real process is more precise and far more interesting. The turtle is not filling its lungs from behind, and the cloaca does not replace ordinary breathing in every situation. It is performing aquatic gas exchange across specialized tissue, supplementing the oxygen obtained when the animal surfaces and breathes air. The ability is especially developed in species adapted to clean, flowing, oxygen-rich rivers. Studies of the white-throated snapping turtle found that the cloacal bursae accounted for nearly half of its oxygen uptake from water, while the Fitzroy River turtle is also highly dependent on this form of respiration during long dives. Cold water can make the strategy even more useful, because cooler conditions slow the turtle's metabolism while often allowing the water to hold more dissolved oxygen. The animal then requires less energy and can remain submerged longer without exposing itself at the surface. Yet this adaptation has limits. Warm, stagnant, polluted, or oxygen-poor water reduces how much oxygen can be absorbed, and even a highly specialized turtle must eventually surface if its needs exceed what the water can provide. This dependence also makes river modification dangerous. Dams, sediment, and declining water quality can damage not only the turtle's habitat, but the invisible respiratory conditions on which its survival depends. What seems like an amusing anatomical curiosity is therefore a carefully tuned relationship between blood, water, temperature, and landscape. The turtle does not merely live in the river as you might live beside one. Part of the river passes rhythmically through its body, delivering oxygen across tissues hidden near the tail. Its breathing is not confined to a chest rising in air. It extends into the moving water around it, making the boundary between animal and habitat quieter and more intimate than it first appears. The smell of rain comes, partly from molecules made by soil bacteria. The first breath after rain is partly the scent of bacterial chemistry rising from the earth. That familiar earthy aroma is often called petrichor, but it is not produced by one substance or one process alone. plant-derived oils accumulated during dry weather, minerals, moisture, and several airborne compounds may all contribute. Yet one of the most recognizable ingredients is geosmin, a powerful smelling molecule made by microorganisms, including soil-dwelling bacteria in the genus Streptomyces. These bacteria spend much of their lives growing as branching filaments through soil and can produce geosmin during stages associated with spore formation. Human noses are extraordinarily sensitive to it, detecting amounts so small that the molecule can be noticed even when only a trace has entered the air. Before rain, much of the geosmin remains trapped among soil particles. When a drop strikes porous ground, it can capture tiny pockets of air beneath itself. These pockets form bubbles that rise through the droplet and burst at its surface, throwing microscopic aerosol particles upward. Those particles can carry geosmin and other material from the soil into the air, where a passing breeze delivers them to your nose. Light or moderate rain can sometimes produce this effect especially well, because droplets striking at suitable speeds create abundant aerosols, whereas violent rain may quickly saturate the ground and change the release process. The smell is therefore not simply water possessing an odor. Pure rainwater is nearly scentless. What you recognize is the atmosphere suddenly carrying substances that were already waiting in plants, rocks, and microbial communities below. Geosmin also has an ecological life beyond human enjoyment. Research suggests that it can influence animals in different ways, attracting some soil arthropods that help disperse bacterial spores while acting as a warning or repellent in other relationships. It can also create earthy flavors in drinking water and fish, showing that a molecule pleasant in a forest can become unwanted elsewhere. There is no firm evidence that humans love petrichor because of one specific ancestral survival adaptation, so that tempting story should remain a possibility rather than a fact. What is certain is already enough to change the moment. When rain darkens a dry path and the air suddenly seems richer, you are detecting the work of organisms too small to see and physics too fast to notice. A falling drop strikes the ground, bubbles form and burst, invisible particles rise, and a bacterial molecule enters your awareness as memory, calm, or longing. The scent feels like the sky has changed, yet much of it has traveled upward from the living soil beneath your feet. Freshly cut grass releases chemical distress signals. That pleasant smell of a freshly mown field begins at the exact moment millions of plant cells are torn open. Grass stores much of its chemistry inside organized membranes and compartments, but cutting crushes this structure and brings enzymes into contact with fatty molecules they could not previously reach. Within seconds, a reaction pathway begins producing a mixture known as green leaf volatiles, including several 6-carbon aldehydes, alcohols, and esters. Compounds such as hexanol and hexanol create much of the sharp green scent you associate with summer lawns. Calling this odor a distress signal is partly useful because the chemicals are released in response to damage and can participate in protection. But the phrase should not be taken to mean that grass feels pain, cries out consciously, or experiences fear. A plant has no brain or nervous system like an animal's. What it does possess is a highly responsive network of cells able to detect broken tissue, water loss, pressure changes, invading microbes, and chemicals associated with feeding insects. The first burst of odor includes material released directly from damaged cells, while later emissions result from active biochemical responses in tissues that remain alive. Some volatile compounds can slow the growth of bacteria or fungi around a wound, influence the behavior of insects, or help coordinate defenses in undamaged parts of the same plant. In natural grazing systems, certain blends may also attract predators or parasitoids that attack the herbivore responsible for the injury. A mechanically mown lawn, however, creates damage on a scale and pattern unlike a single caterpillar bite, so the resulting chemical blend does not carry one simple message with a fixed translation. Different grass species release different proportions of compounds, and temperature, sunlight, moisture, wound size, and the time since cutting all influence what enters the air. Field studies have shown that volatile emissions rise sharply during mowing and can continue afterward as the cut vegetation dries. On a large enough scale, these emissions can even participate in atmospheric chemistry, although a single garden is not creating a major environmental crisis each time it is trimmed. The deeper lesson lies in how differently the same event can be experienced. To you, the smell may signal cleanliness, childhood, or the beginning of summer. To the grass, the chemistry begins because protective barriers have been destroyed and exposed tissue must respond. The fragrance is neither a scream nor a perfume deliberately made for human pleasure. It is the airborne trace of membranes breaking, enzymes awakening, and living cells attempting to manage sudden damage. You inhale it as nostalgia because your brain supplies the meaning, while the plant produces it as part of the difficult work of remaining alive after the blade has passed. The metallic smell on your hands comes from your skin, reacting with metal. The coin in your palm may seem to leave its own smell behind, yet the odor reaching your nose is being manufactured largely by your skin. Solid metals such as iron and copper release almost no vapor at room temperature, so the familiar metallic scent is not simply a cloud of metal atoms drifting into the air. When your fingers touch an iron object, moisture and salts in sweat help release reactive iron ions from its surface. Those ions interact with oily compounds and lipid peroxides already present on your skin, rapidly breaking them into smaller airborne molecules that your nose can detect. Researchers have identified a mixture of aldehydes and ketones in this reaction, including one compound with an especially powerful metallic and mushroom-like odor. Because the human sense of smell can detect some of these chemicals at extremely low concentrations, only a tiny reaction is needed to make your hands seem strongly metallic. Similar chemistry helps explain why blood on skin can produce a related scent. Iron in hemoglobin can participate in reactions with skin oils, although the complete smell of blood also contains many other biological compounds. Different objects may produce slightly different odors because their surfaces, coatings, corrosion, and metal mixtures are not identical, and the oils on one person's skin may differ from those on another. Washing removes much of the odor not because it scrubs away a layer of invisible metal perfume, but because it removes the volatile reaction products and the skin oils from which they formed. The distinction seems small, yet it changes what you are actually experiencing. You believe you are smelling the object, while in reality the object has triggered your own body to create the scent. Touch becomes chemistry. Chemistry becomes airborne information, and your nose interprets the result as a property of the metal. The smell does not belong entirely to your hand or to the object. It appears only when the two meet, a temporary signature created at the boundary between your skin and the material you touched. Bananas are naturally slightly radioactive. A banana can produce a genuine radiation signal without being dangerous, contaminated, or altered by anything humans have done. The reason is potassium, an element every living cell requires to control fluid balance, transmit nerve signals, and help muscles contract. Most potassium atoms are stable, but a very small natural fraction exists as potassium-40, a radioactive form that has been present on Earth since before the planet formed. Because bananas contain a useful amount of potassium, they also contain a tiny quantity of potassium-40, and each decay releases radiation as the unstable atom transforms. Sensitive instruments can detect this activity, which is why bananas are often used as a memorable example of natural radioactivity. One banana is sometimes assigned a banana equivalent dose of about 1/10 of a microsievert, but this is an educational comparison rather than a formal medical unit, and it should not be interpreted as though every banana permanently adds another fixed layer of radiation to your body. Your body already contains radioactive potassium because potassium is present throughout your tissues, and healthy kidneys carefully regulate its total amount. When more potassium enters through food, the body generally removes a corresponding amount over time, keeping the internal concentration within a narrow range. The radiation exposure associated with eating a banana is therefore extremely small compared with the natural background radiation you receive continually from the ground. buildings, the atmosphere, cosmic rays, and even other atoms inside your own body. Bananas do not become unsafe because a radiation detector can notice them, and ordinary consumption does not require special precautions. What the fruit reveals is that the word radioactive does not automatically mean artificial, poisonous, or catastrophic. Radioactivity is a natural property of certain atoms, and those atoms are woven into soil, water, food, and life itself. The banana on a kitchen table looks completely ordinary because it is ordinary. The surprising part is that ordinary matter was never as chemically silent as it appeared. Even within something soft, sweet, and familiar, a small number of ancient atoms are quietly changing into other atoms, one decay at a time. Brazil nuts can contain more natural radioactivity than most foods. Deep within an ordinary, Brazil nut can be a concentration of naturally radioactive material noticeably higher than that found in most foods. Unlike the radioactivity of bananas, which is associated mainly with potassium-40, Brazil nuts are known for accumulating radium isotopes from the ground in which their enormous trees grow. Radium occurs naturally in soils and rocks as part of the long decay chains of uranium and thorium. Chemically, it resembles elements such as barium and calcium, so plant roots can absorb it along with other minerals and transport some of it into developing seeds. The amount is not uniform. Radioactivity can vary greatly between nuts and harvest regions because Amazonian soils differ in geology, mineral content, root conditions, and the availability of radium to the tree. Measurements have found that Brazil nuts may contain substantially more radium than many other common foods, which is why they frequently appear in discussions of naturally radioactive diets. This does not mean that a few Brazil nuts glow, feel warm, contaminate nearby food, or expose the eater to anything resembling a medical scan. The activity remains low in everyday terms, and the radiation cannot be detected by human senses. Researchers are still refining estimates of how much ingested radium becomes biologically available, since some may pass through the digestive system without being absorbed. Long-term dose estimates therefore depend on the measured nuts, the amount eaten, and assumptions about absorption. Radioactivity is also not the only reason to avoid treating Brazil nuts as an unlimited snack. They contain exceptionally high and highly variable amounts of selenium, an essential nutrient that can become harmful when consumed excessively over time. A small serving can provide more than the daily requirement, so eating large quantities regularly is unnecessary regardless of radiation. The truthful lesson is neither that Brazil nuts are dangerous objects, nor that natural radiation can always be ignored. It is that food grows from geology. The minerals drawn through roots carry the history of the rocks below, and some of those minerals contain unstable atoms. When you eat a Brazil nut, you are tasting sunlight stored by a tree, nutrients gathered from soil, and a barely measurable trace of the radioactive processes that have been unfolding beneath the forest for billions of years. Lightning can heat the air to temperatures hotter than the sun's surface. For a fraction of a second, the narrow path of a lightning bolt becomes one of the hottest places you could encounter in Earth's atmosphere. As electrical charge builds within a storm, the insulating air eventually breaks down and becomes ionized, allowing a powerful current to move through a thin channel of plasma. The air inside that channel can briefly reach temperatures close to 30,000 degrees Celsius, roughly 5 times hotter than the visible surface of the sun. This comparison is scientifically accurate, but it can also be misleading, unless you consider scale and duration. Lightning does not contain more total heat than the sun, nor does an entire thundercloud reach such a temperature. The extreme heating is concentrated in a channel only a few centimeters across, and it lasts for an extraordinarily short time. The sun's surface, by contrast, remains hot across an immense area and continually releases vast quantities of energy. Still, the brief atmospheric event is violent enough to transform the surrounding air almost instantly. Molecules are torn apart, atoms lose electrons, and the heated gas expands outward faster than the surrounding air can move aside. This sudden expansion creates a pressure wave that begins as a shock and gradually weakens into the sound you know as thunder. The delay between flash and sound occurs because light reaches you almost immediately, while sound travels much more slowly through air. A nearby strike may produce a sharp crack because you hear a short section of the channel almost at once, while a distant or branching bolt produces a long rumble as sound from different parts reaches you at different times. Lightning can also melt sand into branching glass structures called fulgurites, damage trees by rapidly heating water inside them and ignite fires. Yet most of the air along its path cools quickly once the current ends. What appears from a window as a thin white line is therefore not merely light crossing the sky. It is a temporary electrical pathway in which ordinary air becomes plasma, expands with explosive speed, and then vanishes before your eyes can understand the violence that just occurred. Thunder is the part that arrives later, carrying the physical consequence of heat you could never safely approach. Volcanic eruptions can generate their own lightning. An erupting volcano does not always need a thunderstorm overhead to fill the sky with lightning. Under the right conditions, the rising ash cloud can build its own electrical storm from within. During an explosive eruption, magma is shattered into fragments of rock, glass, and ash, while hot gas drives the material upward at enormous speed. Near the vent, particles collide, scrape, fracture, and separate, transferring electrical charge between surfaces. Smaller and larger particles may carry different charges and move through the plume at different speeds, allowing positive and negative regions to pull apart. Once the electrical difference becomes strong enough to overcome the resistance of the surrounding air, a discharge forms and lightning flashes through the ash. These early sparks can be short, frequent, and concentrated close to the crater, sometimes beginning almost immediately after fragmentation. Higher in the atmosphere, another process can take over. A tall eruption column cools as it rises, and water vapor may form droplets, ice crystals, and hail-like particles. Their collisions can separate charge in ways similar to an ordinary thunderstorm, producing longer flashes farther from the vent. Scientists therefore do not describe volcanic lightning as the result of one universal mechanism. Fracturing magma, colliding ash, particle size, rising convection, water, and ice can all contribute, with their importance changing during different stages of the same eruption. Not every volcano produces visible lightning, and glowing photographs do not mean that lava itself is shooting electricity into the sky. Explosive, ash-rich eruptions are especially favorable because they provide dense clouds of rapidly interacting particles. Lightning detection has become useful for more than dramatic imagery. Networks and satellites can sometimes identify electrical activity above remote or cloud-covered volcanoes, helping observatories confirm that an ash-rich plume is developing when direct visibility is poor. During the enormous 2022 Fungi eruption, researchers recorded nearly 200,000 flashes, with the rate briefly exceeding 2,600 flashes per minute, showing how intensely an eruption plume can electrify. Yet even smaller discharges reveal the same underlying truth. Volcanic ash is not passive smoke. It is fractured matter rising, cooling, colliding, sorting itself by size, and building invisible fields until the air can no longer contain them. The mountain seems to produce fire from below and weather from above, but both displays begin within the same turbulent column of rock and gas. The Sahara Desert was once covered with lakes, rivers, and grasslands. Walk across the Sahara today, and it is difficult to imagine boats moving across its interior, animals grazing beside fresh water, or human communities living far from the few surviving oases. Yet during the African humid period, especially between roughly 11,000 and 5,000 years ago, large parts of North Africa were dramatically wetter than they are now. The entire Sahara was not one continuous tropical jungle, and every region did not become green at exactly the same time, but vast areas that are now hyperarid supported grasslands, wooded savannas, wetlands, rivers, and permanent or seasonal lakes. Scientists know this from many independent records. Beneath desert sand, they have found ancient lake sediments, freshwater shells, fish remains, pollen from vegetation, abandoned river channels, and groundwater left from wetter climates. Rock art portrays cattle, giraffes, elephants, swimmers, and other scenes that would be impossible in much of the modern desert. Archaeological sites reveal people fishing, hunting, gathering plants, and later herding animals in landscapes that offered far more water and food than the Sahara provides today. The transformation was driven largely by slow changes in Earth's orbital orientation. Thousands of years ago, Northern Hemisphere summers received stronger sunlight, heating the land and strengthening the West African monsoon. Moist air traveled farther north, bringing rainfall deep into regions now dominated by dust and dunes. Vegetation then helped reinforce the wet climate. Darker plant-covered ground absorbed more sunlight than bright desert, while reduced dust and increased soil moisture influenced clouds and atmospheric circulation. Lakes expanded, river systems reawakened, and one ancient waterway in Western Sahara may have carried runoff toward the Atlantic through a vast network now buried beneath sediment. The humid era did not end everywhere in a single sudden collapse. As orbital conditions gradually changed, monsoon rainfall weakened, but vegetation loss, dust, regional geography, and periods of drought caused different areas to dry at different rates. By around 5,000 years ago, much of the Green Sahara had moved toward the desert conditions familiar today, and human populations increasingly concentrated near dependable water, including the Nile Valley. The Sahara, therefore, is not a landscape with one permanent identity. Its sand contains evidence of repeated humid periods stretching far deeper into the past, each paced partly by astronomical cycles. The silent desert you see now is only its current state, resting over old shorelines, buried channels, and the remains of lives once organized around water where almost none can now be found. Antarctica is the largest desert on Earth. The largest desert on Earth is not covered in burning sand, but buried beneath ice that can be several kilometers thick. A desert is defined mainly by how little precipitation it receives, not by how hot it becomes, and Antarctica is extraordinarily dry. The continent covers nearly 14 million square kilometers, making it larger than the Sahara, while much of its high interior receives only a tiny amount of new snowfall each year. Some central regions receive the water equivalent of only a few millimeters of precipitation, conditions as dry as or drier than many famous hot deserts. Antarctica can still hold an immense ice sheet because the snow that does fall rarely melts. Layer after layer accumulates across thousands of years, compressing older snow into dense glacial ice. Cold air also holds very little water vapor, and the continent's great elevation helps keep its interior even colder. Mountains force moisture from approaching air, while powerful downslope winds can sweep loose snow away and allow ice to pass directly into vapor through sublimation. Coastal areas receive more snowfall than the interior, so Antarctica is not equally dry everywhere, but the continent as a whole remains a polar desert. Its whiteness can therefore deceive you. You see enough frozen water to raise global sea level dramatically if it melted, yet the atmosphere above much of that ice provides remarkably little new moisture. This is not a contradiction. The ice is a savings account built over an enormous span of time, not evidence of generous yearly snowfall. The landscape reveals that abundance and supply are not the same thing. Antarctica possesses a vast reserve of water precisely because the cold preserves nearly everything that slowly arrives, turning one of the driest places on Earth into the location of its greatest mass of ice. Rivers and lakes of dense brine exist on the ocean floor. Far below the waves, a submarine may approach what looks like the shoreline of a lake, even though it is already surrounded by ocean water. These strange formations are brine pools, bodies of water so heavily loaded with dissolved salt that they become much denser than the seawater above them. In places such as the Gulf of Mexico, ancient seas once evaporated and left enormous deposits of salt beneath layers of sediment. Geological pressure later deformed those deposits, creating domes, cracks, and pathways through which seawater could circulate and dissolve the buried salt. When the concentrated brine emerges from the seafloor, it sinks instead of mixing quickly, flowing downhill through channels and gathering in depressions like underwater rivers and lakes. A visible boundary can form between the brine and ordinary seawater because the two fluids differ in density and chemistry, producing ripples and reflections that resemble a surface seen in open air. Yet entering such a pool would not be like swimming into another harmless lake. Some brines contain very little oxygen and may carry methane or hydrogen sulfide, making their interiors lethal to many animals that cross the boundary. Dead fish and crustaceans have sometimes been observed near these pools, which has inspired dramatic names such as Jacuzzi of Despair. But not every brine pool has identical chemistry or biological effects. Around their edges, highly specialized communities can thrive. Mussels and tube worms may depend on bacteria that obtain energy from methane or sulfur compounds rather than sunlight, allowing food webs to form in permanent darkness. Salt-loving microbes can survive where most familiar organisms cannot, turning a hostile chemical boundary into a rich habitat. These formations teach you that liquids do not automatically blend simply because they touch. Density can preserve a boundary, geology can create a river beneath another body of water, and life can gather beside conditions that would poison it only a short distance away. The ocean floor is not merely wet ground beneath a uniform sea. In some places, it contains its own shorelines, currents, basins, and lakes, hidden inside the larger ocean. The longest mountain range on Earth lies beneath the ocean. If every ocean were suddenly removed, the most extensive mountain system on the planet would appear as an enormous volcanic seam winding between the continents. The global mid-ocean ridge stretches for nearly 65,000 kilometers, far longer than any mountain chain on land, with more than 90% of it concealed beneath deep water. It is not one perfectly unbroken wall or a row of isolated peaks. It is a connected network of ridges, valleys, faults, and volcanic segments running through the Atlantic, Pacific, Indian, Arctic, and Southern Oceans. These mountains form where tectonic plates slowly move apart. As the crust separates, hot material rises from Earth's mantle, partially melts, and produces magma that enters the opening. Much of that magma cools into dark volcanic rock, creating new oceanic crust and pushing older seafloor away from the ridge. This process, called seafloor spreading, continually renews the ocean basins. It does not happen at the same speed everywhere. Some ridges spread slowly and develop steep central valleys, while faster spreading sections can form smoother, broader rises. Earthquakes and volcanic eruptions occur along the system, but most happen far from human sight beneath kilometers of water. Seawater also enters cracks in the young crust, becomes heated by rock and magma, and may return through hydrothermal vents carrying dissolved minerals. Around some vents, microorganisms use chemical energy to support ecosystems that do not depend directly on sunlight, while mineral-rich fluids gradually build chimney-like structures on the seafloor. Scientists recognized the true scale of the ridge only after sonar mapping revealed the hidden topography of the oceans, and magnetic measurements showed matching bands of crust on opposite sides, evidence that new seafloor had been forming and moving outward. The continents on a classroom map may seem like the dominant features of Earth, while the oceans appear as empty blue spaces between them. Beneath that blue, however, the planet is constructing mountains, opening basins, recycling heat, and recording the movement of tectonic plates in newly formed stone. The longest mountain range is largely invisible not because it is small, but because the ocean has hidden one of Earth's greatest structures in plain sight. Crystals larger than cars have grown inside natural caves. Imagine entering a chamber where the floor, walls, and ceiling are crossed by crystals longer than the vehicle that brought you there. Nearly 300 meters beneath Naka in northern Mexico, miners discovered a natural cave containing some of the largest known crystals on Earth. Enormous transparent beams of selenite, a form of gypsum, with the largest measured examples extending more than 11 meters and weighing many tons. They did not grow during a sudden geological event, nor were they carved into shape by flowing water. For hundreds of thousands of years, perhaps close to 1 million, the chamber remained flooded with hot, mineral-rich groundwater and held within an exceptionally narrow range of temperature. Magma beneath the region heated the surrounding water and rock, while calcium sulfate existed largely in a mineral form called anhydrite. As the underground system cooled to roughly the mid-50s Celsius, anhydrite slowly dissolved and supplied the ingredients needed to form gypsum. The water was only slightly more concentrated than the point at which gypsum could remain dissolved, so relatively few crystals began growing, but those that did faced little competition. Molecules continued attaching to their surfaces at an extraordinarily slow rate, allowing the same structures to expand without being interrupted by rapid cooling, drying strong currents, or large changes in chemistry. The crystals became gigantic not because nature accelerated their growth, but because almost nothing disturbed them. Mining operations later pumped away the groundwater that had protected the chamber, making human entry possible but exposing the crystals to hot air with nearly complete humidity. The environment was so dangerous that explorers required cooling equipment and could remain inside for only limited periods. These structures are sometimes described simply as giant cave crystals, yet they are not quartz, diamonds, or frozen glass. Gypsum is a relatively soft mineral containing water within its crystal structure, and once removed from the stable flooded conditions in which it formed, its surface can gradually deteriorate. Their size, therefore, records a balance that no longer fully exists. Steady heat, the correct minerals, deep water, and almost unimaginable patience. You often think of growth as something driven by speed, abundance, and constant activity, but nature teaches the opposite lesson. Under the ground, in a chamber no eye could visit, matter built monuments larger than machines by changing so slowly that a human lifetime would have revealed almost no movement at all. Solid rock can bend and fold without breaking. A cliff may look like the definition of rigidity, yet some cliffs contain layers that have been folded almost like fabric without shattering into separate pieces. Rock can behave in very different ways depending on temperature, pressure, mineral composition, water content, and how quickly force is applied. Near Earth's surface, where temperatures and surrounding pressure are relatively low, sudden stress commonly produces brittle deformation, cracks open, blocks slide along faults, and earthquakes release stored energy. Deeper within the crust, however, rock is confined by the weight of everything above it and warmed by heat rising from Earth's interior. The pressure makes it harder for fractures to open, while the higher temperature allows minerals to change shape through processes that are almost invisible on a human timescale. Imperfections within crystal structures can migrate. Grains can recrystallize into new shapes, atoms can slowly diffuse, and mineral material can dissolve where pressure is greatest before being deposited elsewhere. Individual grains may deform only slightly, but when these changes accumulate across millions of years, entire rock layers can shorten, thicken, stretch, and curve into enormous folds. This does not mean that a slab of stone becomes soft enough to bend in your hands, and it does not require the whole layer to have once been liquid. Geological folding usually occurs while the material remains solid, but solid does not mean incapable of permanent change. A slow force sustained for an immense period can produce an outcome that the same rock would resist under a sudden blow. Different minerals also respond differently, so a sequence of strong and weak layers may buckle, slide, or concentrate strain in complex patterns. Water inside microscopic pores and along grain boundaries can assist chemical reactions and reduce resistance, while the direction of tectonic compression determines how the folds develop. Later uplift and erosion may expose those once deep structures at the surface, allowing you to stand beside stone that preserves the pressure of vanished mountain building events. What appears to be a frozen wave in a canyon wall is not evidence that gravity briefly stopped or that the rock melted and hardened again. It is the remaining shape of motion performed too slowly for any living observer to watch. The fold asks you to reconsider what rigidity means when time is allowed to become part of the force. On the scale of a moment, rock is hard. On the scale of a planet, even stone can flow without ever becoming liquid. Earth's inner core is slowly growing as the planet cools. At the center of Earth, a solid sphere is gradually forming from an ocean of metal that no drill could ever reach. The inner core lies more than 5,000 kilometers beneath your feet and consists mainly of iron, with nickel and smaller amounts of other elements, compressed by pressures millions of times greater than those at the surface. Its temperature is comparable to the visible surface of the sun, yet it remains solid because the crushing pressure raises the melting point of iron. Surrounding it is the liquid outer core, where molten metal flows and carries heat away from the deeper interior. As Earth loses heat over geological time, conditions at the boundary between the liquid and solid cores allow some outer core material to crystallize onto the inner core. The boundary therefore moves slowly outward, and the solid sphere grows. Although scientists cannot state one simple annual rate with certainty because its history depends on the core's composition, thermal conductivity, pattern of cooling, and the difficult question of how the first stable crystals formed, no instrument has photographed this growth. Researchers infer it from earthquake waves passing through the planet, laboratory experiments performed at extreme pressure, and physical models of how iron alloys behave. The freezing process matters far beyond the size of the inner core. When iron-rich material solidifies, it releases heat and tends to leave lighter elements behind in the liquid outer core. The warmer, chemically lighter fluid rises, while cooler material sinks, helping sustain the churning motion of electrically conducting metal. Together with Earth's rotation, that motion powers the geodynamo responsible for most of the planet's magnetic field. The field that guides compasses and helps shield the atmosphere from charged particles is therefore linked partly to metal freezing in darkness at Earth's center. Scientists continue debating the age of the inner core, with estimates ranging widely because new research on heat flow and the difficulty of crystal nucleation can change the reconstruction of its past. It is probably much younger than Earth itself, having begun only after the center cooled enough for solidification to overcome the conditions favoring liquid metal. Even now, the process is neither perfectly uniform nor completely understood, yet the larger picture is clear. Earth is not merely cooling like an abandoned stone. Its loss of heat drives changes, separates materials, moves liquid metal, and gradually builds a solid world inside the world you inhabit. Far below every quiet landscape, the planet is still becoming something new, adding crystal to its hidden center, while the consequences rise outward as a magnetic field surrounding the entire Earth. Earth's magnetic poles have reversed many times. A compass carried into Earth's distant past would sometimes point in the direction you now call south. The planet's magnetic field is generated mainly by the movement of electrically conducting liquid metal in the outer core. But this vast natural dynamo does not produce a perfectly steady bar magnet with permanently fixed ends. Flow patterns within the core continually change, causing the magnetic field to strengthen, weaken, drift, and occasionally reorganize so completely that magnetic north and magnetic south exchange places. Scientists discovered this history because cooling lava and accumulating sediments can preserve the direction of the field that existed when they formed. Iron-bearing minerals inside molten rock tend to align with the surrounding magnetic field. Once the rock solidifies, that orientation becomes locked in place like a geological compass needle. By dating sequences of rocks and measuring their preserved magnetism, researchers have reconstructed a long record containing hundreds of full reversals. The intervals between them are highly irregular. Sometimes reversals have been separated by only tens of thousands of years, while at other times the same polarity has persisted for tens of millions of years. The most recent complete reversal, known as the Brunhes-Matuyama reversal, occurred about 780,000 years ago. A reversal would not mean that Earth physically turns over. that the geographic poles move to opposite continents, or that compasses suddenly reverse during a single night. The transition generally unfolds across hundreds or thousands of years, during which the field may weaken and develop several complicated magnetic poles before settling into the opposite orientation. Paleomagnetic evidence indicates that surface field strength can fall greatly during a reversal, but it does not disappear completely, and the atmosphere would continue providing substantial protection from incoming radiation. Claims that every reversal causes global extinctions are unsupported. The geological record shows no reliable connection between polarity changes and mass extinction events. Nor does the present weakening of some parts of the field prove that another reversal is about to begin, because field intensity has varied many times without producing one. The deeper truth is less dramatic but more instructive. Even the invisible structure that guides a compass is not permanent. Beneath the solid ground, moving metal continually redraws the magnetic environment of the entire planet, and the rocks around you preserve earlier versions of that unseen world. North feels like an absolute direction, only because you were born during one temporary chapter of a field whose history has repeatedly turned itself around. The length of a day changes, slightly from one day to the next. Midnight does not arrive after precisely the same amount of planetary rotation every day, even though your clocks are designed to make it appear that way. A civil day is defined as 24 hours, but the real Earth is not a rigid sphere spinning at an unchanging rate. Its atmosphere moves, its oceans circulate, water shifts between land and sea, ice accumulates and melts, and material deep inside the planet changes motion. Whenever mass or rotational momentum is redistributed, the solid Earth adjusts slightly, much as a spinning skater changes speed by moving the arms inward or outward. Strong winds are among the most important causes of short-term variations. When large atmospheric currents, particularly high-altitude jet streams, speed up in the direction of Earth's rotation, the atmosphere gains angular momentum, and the solid planet slows by a tiny amount. When those winds weaken or change, some of that momentum can return. Ocean currents and tides also exchange momentum with the crust, producing daily, monthly, and seasonal variations. These changes are extremely small, usually measured in fractions of a millisecond or a few milliseconds, so you never feel the ground accelerate beneath you, and an ordinary wristwatch cannot reveal the difference directly. Scientists detect it using techniques such as very long baseline interferometry, which compares signals from distant objects in space as Earth rotates, together with satellite and laser measurements. The result is a precise astronomical time called UT1 based on Earth's actual rotation. Atomic clocks, however, measure time through stable atomic transitions and do not slow down when the planet does. This creates a gradual mismatch between uniform atomic time and irregular planetary time, which is why Earth's rotation has historically been considered when managing civil time. Over much longer periods, lunar tides gradually slow Earth, lengthening the average day as rotational energy is transferred through the Earth-Moon system. But the day-to-day variations are dominated by the restless movement of air and water. Even major earthquakes generally change the length of the day by amounts too small to measure clearly, despite dramatic claims that sometimes follow them. The lesson is not that calendars are unreliable, but that timekeeping requires us to smooth a living planet into an ideal schedule. Your clock divides the day into equal seconds, while Earth completes each turn through a different arrangement of winds, currents, pressure systems, and moving mass. The 24-hour day feels like a permanent container surrounding your life, yet beneath that familiar rhythm, the planet is continually arriving a little early or a little late. The moon still experiences earthquake-like tremors. The moon appears frozen in silence, but instruments left there by Apollo astronauts revealed a world that continues to shiver. Seismometers operated on the lunar surface from the late 1960s until 1977 and recorded thousands of vibrations now called moonquakes. they were not all produced by the same process. Some originated deep below the surface and occurred in repeating patterns connected to Earth's changing tidal pull. As the Moon travels around Earth, gravity flexes its interior slightly, repeatedly stressing particular regions and triggering deep tremors hundreds of kilometers below the surface. Other signals came from meteoroids striking the Moon, where there is no thick atmosphere to burn small incoming objects away before impact. A third type appeared near sunrise and sunset, when surface rocks expanded and contracted as temperatures shifted between the extreme cold of lunar night and the intense heat of lunar day. The most scientifically important and potentially hazardous events are shallow moonquakes, which occur closer to the surface and can reach strengths roughly comparable to moderate earthquakes. Some may be connected to faults formed as the Moon slowly cools and contracts. As its interior loses heat, the entire body shrinks slightly, compressing the crust until sections slip along thrust faults and create shaking. The Moon's seismic behavior differs from Earth's because it lacks active plate tectonics and contains far less water. Lunar rock is dry, fractured, and rigid, so seismic energy is not absorbed as efficiently as it is in Earth's warmer, wetter crust. A quake that might fade relatively quickly on Earth can make the Moon vibrate for many minutes, or even hours, almost as though a struck bell continues ringing after the blow. That prolonged motion could matter for future bases, landing systems, slopes, and equipment intended to remain on the surface for years, especially near faults or unstable terrain. It would still be misleading to imagine the entire Moon constantly shaking, or to assume that brief astronaut visits face an overwhelming danger. Strong shallow moonquakes are uncommon, and much remains uncertain because the Apollo seismic network covered only part of the near side and stopped operating decades ago. New instruments are being developed to listen again, including missions intended to measure the far side for the first time. The old idea of the Moon as a completely dead object is therefore too simple. It has no oceans, weather, or moving crustal plates like Earth, but it continues responding to gravity, impacts, temperature, and its own slow loss of internal heat. The footprints left by astronauts may remain undisturbed for ages, yet beneath them, the lunar ground is not perfectly still. A world can look motionless from across space while continuing to settle, tighten, crack, and quietly tremble within. Ice exists on the planet closest to the Sun. The last place you might search for ancient ice is the world that circles closest to the Sun. Yet Mercury hides frozen water in craters where sunlight has not touched the ground for millions of years. Most of Mercury experiences brutal temperature swings because the planet has almost no substantial atmosphere to spread heat from one region to another. Sunlit ground near the equator can become hot enough to melt lead, while darkness allows the surface to cool dramatically. Near the poles, however, the landscape contains deep craters whose floors remain permanently hidden from the Sun. Mercury's rotational axis is tilted only slightly, so the Sun always stays close to the horizon in these regions, and high crater walls prevent its rays from reaching certain depressions. Temperatures inside these permanent shadows can remain low enough for water ice to survive over geological time, even while nearby sunlit slopes are intensely heated. Astronomers first suspected something unusual when radar observations from Earth revealed bright deposits near Mercury's poles. NASA's Messenger spacecraft later strengthened the case by mapping permanently shadowed terrain, measuring hydrogen with its neutron spectrometer, and examining the brightness and darkness of polar surfaces. The combined evidence indicates that some deposits contain relatively clean water ice, while others may be covered by a dark insulating layer rich in carbon-based material. That covering can protect ice from gradual loss, almost like a blanket made from the remnants of comets or asteroids. The water may have been delivered by impacts, released from Mercury's own rocks, or supplied through several processes over a long period. Scientists are still determining how much came from each source. It would be misleading to imagine glaciers stretching across the planet or snow falling from a Mercurian sky. The ice is concentrated in cold traps, hidden inside craters, that preserve conditions entirely different from the blazing terrain only a short distance away. Mercury teaches you that distance from the sun does not decide temperature by itself. Illumination, shadow, atmosphere, surface shape, and the direction of a planet's axis can matter just as much. A world can stand beside the sun and still contain places where heat almost never arrives. The ice survives not because Mercury escapes solar power, but because the geometry of the planet creates small regions that the sun cannot reach, allowing darkness to preserve water on a world defined by light. Titan has lakes and seas made of liquid methane and ethane. Beyond Saturn's rings lies a moon where clouds gather, rain falls, rivers cut through the ground, and shorelines surround broad seas. Yet almost none of that liquid is water. Titan is so cold that water at its surface behaves more like rock, forming a rigid crust, while methane and ethane can remain liquid and circulate through the environment. NASA's Cassini spacecraft used radar to see through Titan's thick orange haze and revealed hundreds of dark lakes and several large seas, concentrated especially around the northern polar region. Some have branching channels flowing toward them, steep shorelines, islands, and shapes that resemble familiar landscapes on Earth. Radar and infrared measurements indicate that these bodies contain mixtures dominated by methane and ethane, along with dissolved nitrogen and other hydrocarbons. The largest, Kraken Mare, covers an area comparable to a major terrestrial sea, while other named bodies such as Ligeia Mare and Punga Mare form part of an entire polar landscape shaped by liquid. Titan also possesses a methane-based weather cycle. Methane evaporates, enters the atmosphere, forms clouds, falls as rain, drains through channels, and gathers again in low regions. Yet this cycle does not mirror Earth perfectly. Titan's weak sunlight, thick atmosphere, low gravity, seasonal changes, and extremely cold surface make every stage operate differently. Rainstorms may be separated by long dry intervals, lakes can expand or retreat with the seasons, and some liquid may drain into porous layers beneath the surface rather than flowing openly across the ground. Ethane is produced when sunlight alters methane high in the atmosphere, and it can accumulate in lakes because it evaporates less readily. Scientists have also detected signs that some smaller northern lakes may sit within deep-sided depressions, perhaps formed when liquid dissolved surface material in a process loosely comparable to the formation of limestone caves and sinkholes on Earth. None of this proves that Titan's seas contain life, and familiar organisms could not simply swim in them. Liquid methane is chemically and thermally unlike water, and Titan's surface is far too cold for known Earth-like biology to function normally. Still, the moon expands your understanding of what a living-looking landscape can be. Rivers do not require water, clouds do not require warmth, and a shoreline is not uniquely terrestrial. From a distance, Titan appears wrapped and featureless beneath its haze, but below that veil lies a world where weather has chosen another substance to carry out the work that water performs here. It asks you to separate the pattern from the material. Perhaps what makes a planet seem familiar is not what flows across it, but the way that flowing substance connects sky, land, and time. Earth collects thousands of tons of cosmic dust every year. While you move through an ordinary day, material older than Earth is quietly settling onto rooftops, oceans, forests, ice sheets, and the clothes of people who will never notice it. Our planet continually encounters tiny grains released by comets and by collisions between asteroids, most of them far smaller than the meteorites displayed in museums. As these particles enter the atmosphere, frictional heating and collisions with air molecules alter many of them. Some vaporize completely, adding their elements to the upper atmosphere. Others partially or fully melt and cool into microscopic rounded grains called cosmic spherules, while the slowest and most fragile arrivals may survive with much of their original structure intact. Measuring the total amount is difficult because extraterrestrial grains become mixed with overwhelming quantities of ordinary dust from soil, industry, volcanoes, fires, and the ocean. To reduce this contamination, researchers spent years collecting exceptionally clean snow near Concordia Station at Dome Sea in Antarctica, where snowfall is low, human activity is limited, and particles can accumulate within carefully dated layers. By extracting and identifying thousands of unmelted micrometeorites and melted cosmic spherules, they estimated that roughly 5,200 tons of extraterrestrial dust in the studied size range reach Earth's surface each year. With a substantial uncertainty around that number, their calculations suggested that a larger quantity, around 15,000 tons annually, approaches Earth before atmospheric entry, meaning much of the incoming material is destroyed or transformed before reaching the ground. Estimates from other methods can differ because they include different particle sizes and measure different stages of the journey. So, thousands of tons is more scientifically honest than pretending there is one perfectly known total. Much of the dust appears to originate from comets, with asteroids supplying another share. Each individual grain is almost weightless, but the global accumulation becomes enormous because the process continues across the entire planet, day and night. These particles are scientifically valuable because some preserve minerals, carbon-rich material, and chemical clues from the early solar system. By studying them, researchers can investigate the bodies from which they came and the changes they experienced while entering the atmosphere. Cosmic dust may also have delivered useful carbon compounds and other materials to the young Earth, though scientists should not claim that dust alone created life. The striking truth is simpler. Earth is not a closed world traveling through empty space. It moves through a thin and continual rain of debris, collecting fragments of comets and asteroids grain by grain. Most will vanish into mud, seawater, or household dust without ever being recognized. Yet together they form an almost invisible exchange between the planet and the wider solar system. The ground beneath you contains not only material shaped by Earth, but tiny arrivals that completed journeys through interplanetary space before becoming part of this world. Sunlight can take thousands of years to escape the sun. The sunlight, resting on your skin today, began its journey long before you, your parents, or perhaps even your civilization existed. Deep inside the sun, hydrogen nuclei fuse and release energy. But that energy cannot simply fly straight towards space because the solar interior is unimaginably dense. Light is repeatedly absorbed, scattered, and re-emitted by charged particles, changing direction so often that its outward journey resembles a traveler taking countless tiny, uncertain steps. Scientists estimate that energy moving through the sun's radiative zone may take around 170,000 years to reach the upper layers, though the exact time depends on how the process is modeled. It is also important to understand that the photon eventually leaving the surface is not necessarily the original photon created in the core. The energy has been transferred and transformed many times along the way. Nearer the surface, rising and sinking currents of hot plasma carry it farther outward until it finally escapes from the visible layer of the sun. Only then does the journey become fast and direct, with the light crossing the space between the sun and Earth in a little over 8 minutes. So when morning light enters your room, you are not simply seeing something produced 8 minutes earlier. You are receiving energy whose long passage began deep within the sun during a chapter of human history that may now be almost entirely forgotten. The brightness feels immediate, yet its origin is ancient, reminding you that even the most familiar moment can be the final arrival of a journey that lasted thousands of human lifetimes. Cosmic rays can, silently, change data inside computers. A computer can make a mistake even when its hardware is working exactly as designed. High above you, energetic particles known as cosmic rays continually arrive from the sun and from violent events far beyond the solar system. And when they strike Earth's atmosphere, they produce showers of secondary particles that can reach the ground. Occasionally, one of these particles passes through a computer chip and releases enough electrical charge to alter the state of a memory cell. A stored 0 may become a 1, or a 1 may become a 0, creating what engineers call a single event upset, or more simply, a bit flip. The chip is not permanently damaged, and there may be no visible sign that anything happened, yet one tiny piece of information has changed. Most such errors are harmless or corrected automatically, but in the wrong location, a flipped bit could corrupt a calculation, alter stored data, crash a program, or interfere with the operation of a spacecraft. The problem becomes more serious at high altitudes and in space, where the atmosphere provides less protection, but terrestrial computers are not completely immune. Engineers reduce the risk with error-correcting memory, duplicated calculations, shielding, and systems that check whether stored information still makes sense. This does not mean every unexplained computer failure was caused by a particle from space, and cosmic rays should not become a convenient explanation for ordinary software bugs. What makes the phenomenon remarkable is that digital information, which appears abstract and untouchable on a screen, must always be held in physical matter. Every photograph, number, and sentence in memory depends on tiny electrical states inside silicon, and a particle traveling across the universe can sometimes disturb one of them. The event may last less than a blink, but it reveals that even the quiet logic of a machine remains connected to the restless physical universe surrounding it. Some distant planets have iron rain falling from their skies. On a planet where daylight is hot enough to vaporize metal, rain becomes something far stranger than water. WASP-76b is an enormous gas giant, orbiting so close to its star that it is thought to keep one hemisphere facing the star while the opposite side remains in permanent darkness. Temperatures on the day side rise above 2,400 degrees Celsius, allowing iron to exist as vapor in the atmosphere rather than as a solid object you could hold. Powerful winds can carry this iron-rich gas toward the cooler boundary of the night side, where the temperature falls enough for the iron to condense. Astronomers studying the planet's atmosphere detected iron vapor on one side of the planet but not on the other, leading them to infer that the missing iron may be condensing and falling into the darker atmosphere as droplets. No spacecraft has watched this rain directly, so the phrase iron rain describes the strongest interpretation of atmospheric measurements rather than a photographed weather event. Even so, the physics behind it is realistic. Weather depends on which substances can evaporate and condense under local conditions, and water is only one possible material. On Earth, our temperature range makes liquid water the familiar substance that moves through clouds and storms. On far hotter worlds, clouds may contain vaporized rock or metal, and the changing temperature between hemispheres can create cycles unlike anything in our sky. WASP-76b therefore teaches you that weather is not a universal collection of rain, snow, and wind. It is matter responding to temperature, pressure, gravity, and motion. Change those conditions enough, and something you know as a building material can become part of the atmosphere, cross a planet on violent winds, and fall through an eternal night as metallic rain. The moon is moving away from Earth by several centimeters each year. The moon you see tonight is slightly farther from Earth than it was when you were born, and the reason begins in the oceans rather than in the sky. Apollo astronauts and later lunar missions placed reflective arrays on the moon, allowing observatories on Earth to fire brief laser pulses toward them and measure the round-trip travel time of the returning light. Decades of these measurements show that the moon's average distance is increasing by about 3.8 centimeters each year. This slow separation is driven primarily by tides. The moon's gravity raises broad tidal bulges in Earth's oceans and also deforms the solid planet slightly, but Earth rotates much faster than the moon travels around it. Because of that difference, friction carries the tidal bulges a little ahead of the line directly connecting Earth and the moon. Their gravity then pulls the moon forward along its orbit, transferring a small amount of Earth's rotational energy and angular momentum to the moon. The extra orbital energy moves the moon into a slightly wider orbit, while Earth's rotation slows by a tiny amount, gradually lengthening the average day over geological time. The recession rate has not remained constant throughout history because the arrangement of continents, the depth and shape of ocean basins, and the efficiency of tidal friction have all changed. You therefore cannot simply multiply today's rate backward and reconstruct the entire past of the Earth-Moon system. Geological evidence suggests that the Moon was much closer long ago, when days were shorter and tides could behave very differently. But today's value describes only the current chapter. Nor is the Moon about to escape Earth. Its outward motion is extraordinarily slow compared with its average distance of roughly 384,000 kilometers, and the Sun's evolution will transform the system long before this simple process could continue indefinitely. What matters is the exchange occurring now. Every ocean tide is part of a gravitational conversation in which Earth surrenders an almost immeasurable fraction of its spin, and the Moon responds by moving outward. The night sky seems to return in the same arrangement, yet the partnership above you is never perfectly repeated. The Moon rises from a path that has widened by less than the length of a finger since last year, quietly proving that even the most familiar relationship in the sky is still changing. The upper atmosphere glows, faintly, even on moonless nights. Remove the Moon, distant city lights, and every visible star from the night sky, and the darkness above you would still not become perfectly black. High in Earth's atmosphere, atoms and molecules continuously produce a delicate emission known as airglow, a light so faint that it is usually overwhelmed by brighter objects, but can be recorded by sensitive cameras and occasionally noticed under exceptionally dark conditions. Its energy begins largely with sunlight, even though much of the glow appears on the night side of the planet. During the day, ultraviolet radiation breaks apart oxygen molecules, removes electrons from atoms, and leaves atmospheric particles in energized states. After sunset, those separated pieces continue moving and reacting. Oxygen atoms can reunite, ions can capture free electrons, and excited molecules can release their stored energy as photons while returning to more stable states. Different reactions create different colors at different heights. Atomic oxygen can produce green and deep red emissions. Sodium can contribute yellow light, and hydroxyl molecules create strong bands that extend into the infrared. These layers may lie tens or hundreds of kilometers above the surface, where the atmosphere is extremely thin but still chemically active. Airglow is not the same as an aurora. Auroras are intensified when energetic particles from space are guided by Earth's magnetic field into the upper atmosphere, often forming bright curtains near polar regions. Airglow is much more widespread and persistent, arising from ordinary solar-driven chemistry that continues across the atmosphere every day and night. It is also not perfectly smooth. Winds, tides, weather systems, and atmospheric gravity waves moving upward from lower layers can compress and disturb the glowing gases, producing ripples and bands visible from satellites or long-exposure photographs. By studying those patterns, scientists can trace motion in a region too high for balloons and generally too low for most satellites to sample directly for long periods. The faint glow therefore becomes a diagnostic screen on which the upper atmosphere displays its temperature, density, chemistry, and movement. To your unaided eyes, a moonless night may feel like an absence, a dark space waiting beyond the reach of Earth. Yet the planet does not end where daylight disappears. Above you, oxygen, sodium, nitrogen, and hydroxyl are still exchanging energy, releasing tiny fragments of light into the night. Darkness is not a complete extinguishing of the atmosphere, but a condition in which its quietest chemistry finally becomes almost visible. A teaspoon of neutron star matter would weigh billions of tons. A teaspoon taken from a neutron star would outweigh a mountain, but the truly unsettling part is that such a spoonful could never be carried home and placed safely on a table. Neutron stars form when certain massive stars exhaust the fuel supporting their cores and collapse during a supernova. Gravity compresses the core so violently that electrons are forced into protons, helping create neutrons and releasing enormous numbers of neutrinos. What remains may contain more mass than the Sun packed into a sphere only about the size of a city. Under those conditions, a volume comparable to a teaspoon can contain roughly 1 billion tons of matter, although the exact figure depends on where inside the star the sample is imagined, because density rises dramatically from the crust toward the core. The familiar statement is therefore a scale comparison rather than the description of a possible laboratory object. Near the surface, atomic nuclei may form a rigid crust immersed in a sea of electrons. Deeper down, nuclei become increasingly rich in neutrons, then begin losing neutrons into the surrounding material. Farther inside, matter is compressed beyond anything that can be reproduced for more than an instant on Earth, and scientists are still uncertain whether the deepest core contains mostly neutrons and protons, or more exotic states involving unusual particles or liberated quarks. The star avoids further collapse partly because quantum effects and powerful nuclear interactions resist compression. But if sufficient mass is added, even those forms of support may fail and a black hole can form. A teaspoon removed from the star would also not remain a neat lump of ultra-dense material under ordinary pressure. The extraordinary compression is maintained by the gravity of the entire neutron star. Without that confinement, the material would violently expand and transform. So the phrase, "what it would weigh on Earth," asks you to imagine its mass while temporarily ignoring the conditions required to keep it compact. Weight would also depend on local gravity, while the amount of matter itself is better described by mass. Yet the comparison remains useful because daily language has no intuitive scale for such density. You know teaspoons as containers for sugar or medicine, and neutron stars force that small domestic volume to carry the imagined mass of landscapes. Their density is not simply a large number. It is the final result of a stellar core being stripped of the spacious atomic structure that makes ordinary matter feel solid. The chair beneath you, the ground below it, and your own body are mostly open space at the atomic level. Inside a neutron star, gravity has nearly erased that openness, pressing matter toward nature's most extreme stable form and turning a city-sized remnant into something heavier than the Sun. Some pulsars spin hundreds of times every second. A star no wider than a city can complete hundreds of full rotations during the time it takes your heart to beat once. Pulsars are rapidly rotating neutron stars, the compressed remnants left when certain massive stars exhaust their fuel and their cores collapse. During that collapse, an enormous amount of matter is forced into a remarkably small volume, while the star's original rotation becomes much faster for the same basic reason a spinning skater accelerates when drawing the arms inward. Yet the fastest pulsars usually have an additional history. Many begin in binary systems beside another star, and over millions or billions of years, gas pulled from the companion can spiral toward the neutron star. This falling material transfers angular momentum, gradually spinning the old neutron star faster until it becomes what astronomers call a millisecond pulsar. Some rotate several hundred times each second, and the fastest confirmed example completes more than 700 rotations per second. The word pulsar does not mean that the star itself repeatedly switches on and off. A pulsar possesses an intense magnetic field, and energetic particles near its magnetic poles produce narrow beams of radio waves, X-rays, or gamma rays. Because the magnetic axis is usually tilted away from the rotation axis, these beams sweep through space as the star turns. When one crosses Earth, telescopes detect a pulse, much as you would see repeated flashes from a distant lighthouse whose beam passes over you. If the beam never points toward Earth, the same object may remain invisible as a pulsar even though it continues rotating. The regularity of some millisecond pulsars is so extraordinary that astronomers use them as natural clocks, measuring tiny variations in pulse arrival times to study binary companions. Test gravity, search for gravitational waves, and investigate the motion of matter between stars. They are not perfectly unchanging clocks. Pulsars gradually lose rotational energy. Some experience sudden changes called glitches, and binary motion can shift the timing of their signals. Their speed also has a physical limit, because a star rotating too rapidly would begin losing material from its equator or become unstable. What survives is a balance between crushing gravity, the strength of ultra-dense matter, and motion fast enough to challenge your intuition. When you picture a star, you may imagine a vast glowing sphere turning slowly in darkness. A millisecond pulsar asks you to replace that image with something far stranger. More mass than the sun compressed into a city-sized body, rotating hundreds of times each second, sending a beam across the universe with such precision that human beings can read its rhythm from thousands of light years away. Solar storms can disturb power grids and radio signals on Earth. Disturbance beginning on the sun can cross millions of kilometers of space and arrive at Earth as a problem for a radio operator, a satellite, or an electrical grid. The phrase solar storm describes several related events rather than one single explosion. A solar flare releases an intense burst of electromagnetic radiation, while a coronal mass ejection can throw a vast cloud of magnetized plasma away from the sun. The radiation from a flare reaches Earth at the speed of light and can rapidly alter the sunlit upper atmosphere. High-energy X-rays and ultraviolet light increase ionization within the ionosphere, the region through which many radio signals travel or reflect. This can weaken or completely absorb certain high-frequency transmissions, producing radio blackouts that affect aviation, maritime communication, emergency operations, and other systems that rely on signals traveling beyond the horizon. A coronal mass ejection moves far more slowly, often requiring days to arrive, but it can produce a geomagnetic storm if its magnetic field connects efficiently with Earth's. Energy then enters the magnetosphere, drives electrical currents through the upper atmosphere, and causes Earth's magnetic field to change. Those changing magnetic conditions can induce unwanted currents in long conductors at the surface, including power lines and pipelines. In an electrical grid, these geomagnetically induced currents may disturb voltage control, increase reactive power demand, overheat transformers, or trigger protective systems. The danger is not that sunlight directly enters your home and destroys every electronic device. Most small disconnected appliances are not the primary concern. Large interconnected systems are more vulnerable because their long transmission lines provide pathways through which induced currents can flow. The severity also depends on the storm's strength, the direction of its magnetic field, local geology, grid design, latitude, and the ability of operators to respond. Solar activity can additionally interfere with satellite electronics, navigation signals, and spacecraft operations, while energetic particles can create increased radiation risk for astronauts and some high-altitude flights. Forecasting therefore matters. Space weather satellites observe the sun, and spacecraft positioned upstream of Earth measure the solar wind before it reaches the planet, giving operators time to adjust satellite orientation, delay sensitive operations, redistribute electrical loads, or prepare for communication problems. Most solar storms pass without producing a catastrophe, and dramatic auroras may be their most visible result. But the systems of modern civilization have extended into regions where solar activity can reach them. A radio message and a current moving through a transformer may feel entirely human, engineered, and separate from the sky. Yet both still operate inside a planetary environment connected to the sun, and a magnetic eruption far away can remind you that technology has never stopped being part of nature. The cornea gets oxygen directly from the air because it has no blood vessels. The clear window at the front of your eye stays transparent, partly by refusing something nearly every other living tissue depends upon: blood vessels running through it. The cornea bends incoming light and provides much of the eye's focusing power, so a dense network of vessels crossing its center would scatter light and interfere with vision. Instead, the healthy central cornea is avascular, meaning it contains no blood vessels of its own. Its cells still require oxygen and nutrients, but they obtain them through several surrounding sources. When your eyes are open, Oxygen from the atmosphere dissolves into the thin tear film covering the corneal surface and then diffuses into the tissue. The deeper layers also receive support from the aqueous humor, the clear fluid inside the front of the eye, while vessels around the corneal edge contribute to the peripheral region. Tears therefore do much more than express emotion or wash away dust. They create a smooth optical surface, carry protective molecules, supply dissolved substances, and maintain the moist boundary through which atmospheric oxygen can reach the cornea. When you close your eyes, the oxygen supply from open air decreases. Oxygen then comes partly from blood vessels within the inner eyelid and from the fluids surrounding the cornea, but the available level is lower than when the eye is exposed to the atmosphere. This is one reason contact lens design matters. A lens rests between the air and the tear-covered cornea, and if its material transmits too little oxygen, especially during prolonged or overnight wear, corneal cells can experience hypoxia. Their metabolism changes, acidic byproducts can accumulate, fluid balance may be disturbed, and the cornea can swell or lose some of its clarity. Chronic oxygen shortage and inflammation may even encourage blood vessels to grow inward from the edge, a response that delivers oxygen but threatens the transparency the cornea is meant to preserve. Modern contact lenses are designed with oxygen transmission in mind, yet proper hygiene, replacement schedules, and professional guidance remain important because oxygen permeability does not eliminate the risks of infection or mechanical irritation. The absence of vessels also does not mean the cornea lacks sensation. It contains an exceptionally rich network of nerves, which is why a tiny scratch or grain of sand can cause intense pain and immediate tearing. This creates an elegant biological contrast. The cornea sacrifices its own direct blood supply so that light can pass through clearly, then relies on air, tears, internal fluid, and peripheral circulation to remain alive. Each time you open your eyes, you are not merely allowing light to enter. You are also restoring contact between the atmosphere and a living transparent tissue, letting oxygen cross an invisible wet surface so that the window through which you see the world can continue seeing without being seen. Babies are born with more bones than adults. Before a newborn can stand, walk, or even hold up its own head, its skeleton already contains more separate pieces than the skeleton of the adult it may one day become. Babies are commonly described as being born with around 270 bones, while most adults have about 206, although the exact number varies depending on which small structures are counted and how far development has progressed. The difference does not mean that dozens of bones disappear. Many begin as separate sections of bone or cartilage and gradually fuse as the body grows. The skull provides one of the clearest examples. At birth, its plates are separated by flexible seams called sutures, with wider soft areas known as fontanelles between some of them. This arrangement allows the head to change shape slightly during birth and gives the rapidly growing brain room to expand during early childhood. Over time, the plates enlarge and the gaps narrow, producing a stronger protective structure without becoming one completely solid shell. Similar unions occur elsewhere. The sacrum near the base of the spine begins as several vertebrae that later fuse, and the adult hip bone forms from 3 major parts that are separate during childhood. Some long bones also develop from multiple growth centers with cartilage zones between the shaft and the ends. Cells within these growth plates divide and produce new tissue, which is gradually replaced by bone, allowing the arms and legs to lengthen. When growth is complete, the plates close and formerly separate regions join. This is why the statement that babies have more bones is broadly true but slightly simplified. Some of the structures counted in infancy are partly cartilage, and anatomists may classify developing elements differently. What matters is the process hidden beneath the number. A child's skeleton is not a miniature adult frame that merely increases in size. It is a changing construction made flexible where birth and growth require flexibility, then strengthened through carefully timed fusion. The adult skeleton seems fixed only because most of this quiet joining has already ended. Your body reached solidity not by beginning complete, but by allowing many separate pieces to grow toward one another until the spaces between them were no longer needed. The heart contains its own electrical pacemaker. Your heart does not wait for the brain to issue a command before every beat. Embedded in the upper wall of the right atrium is a small group of specialized cells called the sinoatrial node, often described as the heart's natural pacemaker. Unlike ordinary heart muscle cells, these cells slowly build an electrical charge on their own. Once that charge reaches a threshold, they generate an impulse that spreads across the atria and causes them to contract, pushing blood into the lower chambers. The signal then reaches another small structure called the atrioventricular node, where it pauses briefly. That delay gives the ventricles enough time to fill before the impulse travels through a network of conducting fibers and triggers a powerful contraction from the lower part of the heart upward. This ordered sequence allows the chambers to work as a coordinated pump rather than squeezing randomly. The heart's electrical system is intrinsic, meaning it can continue producing rhythm even without a new instruction from the brain. A heart removed from the body under carefully controlled laboratory conditions can keep beating for a time if it receives oxygen and suitable nutrients. Yet this does not mean the brain has no influence. The autonomic nervous system can speed the pacemaker during exercise, fear, or excitement and slow it during rest. Hormones, body temperature, blood chemistry, medications, and illness can also modify its rate. If the sinoatrial node fails, other parts of the conduction system may generate a slower backup rhythm, although that rhythm may not be adequate or reliable. An artificial pacemaker can then monitor the heart and deliver electrical impulses when necessary. The pulse you feel at your wrist is therefore the distant mechanical result of a microscopic electrical event beginning inside the heart itself. You experience each beat as a single thump, but beneath it lies a precisely timed journey through cells that generate, delay, guide, and distribute electricity. The heart is not independent of the rest of the body, yet it carries its own basic rhythm within its tissue. Long before you notice emotion in your chest or decide to move a muscle, a small cluster of cells has already begun preparing the next beat, maintaining life through a cycle that continues largely outside conscious awareness. Red blood cells can travel around the body in about a minute. A red blood cell pushed from the left side of your heart may travel through distant tissues and return to the heart before a full minute has passed. The exact time is not fixed because circulation speeds up during exercise and slows in different organs and vessels. But at rest, the entire volume of blood can be pumped through the heart roughly once each minute. The journey begins when the left ventricle contracts and sends oxygen-rich blood into the aorta, the body's largest artery. From there, the flow divides again and again through smaller arteries and arterioles until the red blood cell enters a capillary so narrow that it may have to bend and pass almost one cell at a time. Here the movement becomes slower, giving oxygen time to leave the hemoglobin inside the cell and diffuse into nearby tissues. Carbon dioxide and other products can move in the opposite direction. The cell then enters small veins, joins progressively larger vessels, and returns to the right side of the heart. It is pumped toward the lungs, where it passes through another delicate capillary network surrounding the air sacs. Carbon dioxide is released, oxygen attaches to hemoglobin, and the cell returns to the left side of the heart, ready to begin again. It does not follow one identical circular track on every journey. On one pass, it may supply a muscle in your leg. On another, it may move through the kidneys, skin, digestive organs, or brain. Blood flow is continually redirected according to need. During exercise, more is sent toward working muscles and skin, while after a meal, the digestive system receives a larger share. A red blood cell can repeat these circuits thousands of times during its life of roughly 4 months, traveling through vessels of radically different size without a nucleus or the ability to repair itself in the way many other cells can. Its flexible, indented shape provides a broad surface for gas exchange and allows it to squeeze through narrow passages, but repeated mechanical stress eventually wears it down. The spleen and liver remove aging cells, while bone marrow continually produces replacements. What feels to you like a stable body is therefore supplied by ceaseless motion. Even while you sit completely still, millions of red cells are completing journeys that cross the body in less time than it takes to consider them, delivering the oxygen that makes consideration possible and returning to begin again. Your blood vessels could circle Earth more than twice. Hidden beneath your skin is a transport network so extensive that, if every vessel in an adult human body could be removed, straightened, and placed end to end, common estimates suggest it could stretch for around 100,000 kilometers, enough to circle Earth more than twice. This figure is an approximation rather than a measurement that applies equally to every person, because the total depends on body size, age, tissue composition, and exactly which microscopic vessels are counted. But the scale is still extraordinary. Most of that length does not come from the large arteries and veins you can name. It comes from the immense number of arterioles, venules, and especially capillaries branching through nearly every living tissue. The journey begins with thick, elastic arteries built to withstand pressure from the heart. These divide into progressively smaller vessels that regulate where blood is sent, narrowing or widening according to the needs of muscles, organs, skin, and other tissues. Eventually, blood enters capillaries whose walls are often only a single cell thick. Here, oxygen, nutrients, hormones, heat, carbon dioxide, and waste products can move between the bloodstream and surrounding cells across extremely short distances. A capillary may be almost unimaginably small, yet the body contains so many that their combined length dominates the entire vascular system. They are not all filled equally at every moment. Blood is constantly redistributed, with some networks receiving more flow while others receive less, depending on temperature, activity, digestion, stress, and local chemical signals. This arrangement allows a heart positioned in the chest to support cells in the fingertips, eyes, kidneys, and deepest muscles without requiring one enormous open channel running through the body. The network also repairs and remodels itself. New vessels can form when tissues need a greater blood supply, while unused branches may shrink, and damage to vessel walls can trigger clotting, inflammation, or long-term structural change. When disease narrows even a small region, the effect can spread far beyond that location because every vessel is part of a connected pressure and delivery system. The familiar claim about circling Earth is therefore more than an impressive comparison. It reveals how life solves the problem of distance inside a body. No cell can remain useful for long if it is too far from exchange, so the vascular system repeatedly divides until the space between blood and tissue becomes microscopic. You experience yourself as one continuous person, yet that unity depends on an almost planetary web of passageways, folded into the boundaries of a single human form. Every quiet moment is supported by traffic moving through routes you will never see, proving that the body contains distances far greater than its outer size suggests. The surface of the stomach lining replaces itself every few days. Your stomach holds an acid strong enough to damage living tissue. Yet under healthy conditions, it does not simply digest its own walls. The protection begins with a constantly maintained inner surface built from specialized epithelial cells, many of which live only a few days before being shed and replaced. Saying that the stomach lining renews itself every few days is broadly accurate for the rapidly turning over surface mucous cells. But it does not mean the entire stomach disappears and rebuilds at once. The stomach wall contains several layers and many different cell types, each following its own timetable. Along the inner surface are millions of small openings called gastric pits, leading into glands that produce acid, digestive enzymes, hormones, and protective secretions. Stem and progenitor cells located within these glandular regions continually divide. Their descendants migrate toward the surface, mature into mucus-producing cells, perform their protective work, and are eventually lost into the stomach contents. The mucus they release forms a slippery barrier that traps bicarbonate, creating a thin zone near the tissue where acidity is much lower than in the center of the stomach. Tight connections between neighboring cells prevent acid from easily leaking through, while a rich blood supply carries away unwanted hydrogen ions and provides oxygen and nutrients for repair. These defenses must operate continuously because eating, digestion, alcohol, medications, infection, and ordinary mechanical movement can all challenge the surface. When a cell is injured, nearby cells can spread across the damaged area quickly, and deeper stem cells produce longer-term replacements. Some gland cells, such as those that release acid or digestive precursors, survive much longer than the surface cells, so renewal is a carefully organized flow rather than a complete reset. If the protective system becomes overwhelmed, ulcers can develop. Infection with Helicobacter pylori, frequent use of certain anti-inflammatory medicines, impaired blood flow, and other conditions can weaken defenses or intensify injury, allowing acid and enzymes to reach vulnerable tissue. Acid is therefore not the enemy by itself. It is essential for digestion, protein breakdown, and control of many swallowed microbes. The danger appears when production and protection fall out of balance. Your stomach survives by accepting that its most exposed cells are temporary. Instead of asking one surface to endure years of chemical attack, the body continually sends new cells forward, lets them serve briefly, and replaces them before accumulated damage becomes permanent. The lining remains familiar because the pattern continues, even though many of the cells forming it today were not there last week. What seems like a stable wall is actually a moving frontier, preserved through renewal rather than stillness. Taste receptor cells are regularly replaced throughout your life. The cells helping you recognize sweetness, bitterness, saltiness, sourness, and savory flavors are not permanent witnesses to everything you eat. Most taste receptor cells survive for only days or weeks before they are removed and replaced, with many estimates placing their average lifespan near 10 to 14 days. Although different cell types may renew at different rates, these cells are gathered inside taste buds, small sensory structures, located mainly within specialized papillae on the tongue, but also in parts of the mouth and throat. Each taste bud contains several kinds of cells working together. Some detect particular chemicals from food, others support the structure, and still others act as immature replacements. When food dissolves in saliva, its molecules enter tiny openings at the top of the taste bud and interact with receptors or ion channels on exposed sensory cells. The resulting chemical change alters the cell's electrical state and leads to signals being passed to nearby nerve fibers. Those nerves carry information toward the brain, where it is combined with smell, temperature, texture, irritation, memory, and expectation to produce the full experience you call flavor. Because taste cells sit at a boundary exposed to heat, cold, acidity, friction, microbes, and countless food chemicals, long-term survival would be difficult. Basal cells near the taste bud continually divide, and their descendants mature into new receptor cells that connect functionally with existing sensory nerves. This creates an unusual engineering problem. The nervous system must preserve recognizable taste information even while the individual detectors are repeatedly replaced. Renewal is usually seamless, but it can be disturbed. Aging, infections, nutritional deficiencies, smoking, radiation treatment, certain medicines, nerve injury, and inflammation may reduce taste sensitivity or alter the speed and quality of regeneration. A burned tongue may temporarily make food seem dull because surface tissues and receptor cells have been injured, yet sensation often improves as new cells mature and normal signaling returns. Taste renewal does not mean preferences are erased every 2 weeks. Your memories, learned associations, cultural habits, and brain circuits persist far longer than the receptor cells that provide new input. The replacement occurs at the sensory doorway, not in the entire system that interprets what enters. This is why the same food can remain comforting for decades, even though none of the original taste cells that first detected it are still present. Each meal is sampled by a changing population of microscopic receptors, while the mind connects their brief signals to a much longer personal history. The flavor feels continuous, but the biological instruments creating its first electrical trace are temporary, quietly disappearing and being rebuilt so that the world can keep reaching you through taste. Fingerprints begin forming before birth. Long before your hands ever touch the outside world, the patterns that will distinguish your fingers for life are already being drawn in darkness. Fingerprints begin developing during the early months before birth, when the skin on the fingertips is growing over temporary swellings called volar pads. The basic pattern is influenced by genes, which help determine whether the ridges tend to form loops, whorls, or arches. But genes do not specify every tiny fork, ending, and island within the print. Those finer details emerge from a complicated interaction between the rate at which the fetus grows, the changing shape of the fingertip, pressure within surrounding tissues, contact with the uterine environment, and small differences in how the skin stretches. Even identical twins, who share nearly the same DNA, develop different fingerprints because they do not experience every microscopic force in precisely the same way. By roughly the middle of pregnancy, the major ridge patterns are established, and later growth enlarges them without fundamentally redesigning them. The ridges are useful rather than merely decorative. They help the skin resist slipping, improve grip on certain surfaces, and shape the way vibrations travel through the fingertips, supporting fine touch. Their permanence comes from the deeper organization of the skin. Superficial cells are continually shed and replaced, yet new cells follow the same underlying ridge arrangement, allowing a fingerprint to survive ordinary cuts, peeling, and daily wear. Only an injury deep enough to damage the pattern-forming layers may leave a permanent scar that alters the print. Certain rare genetic conditions can also reduce or eliminate ridge formation, showing that fingerprints are biological structures rather than magical signatures. What makes them remarkable is the balance between inheritance and accident. Your genes helped prepare the landscape, but countless unrepeatable conditions completed the design. Before you had memories, choices, or even a name, your fingertips had already recorded a history of development that no other person experienced in exactly the same way. The pattern used later to identify you was created before you could identify anything at all. Bone marrow produces millions of new blood cells every second. Inside several of your bones, a soft tissue that looks nothing like a factory is producing new blood cells at a rate that ordinary machinery would struggle to match. Bone marrow contains hematopoietic stem cells, rare cells capable of renewing themselves while also giving rise to the many specialized cells circulating through your blood. In a healthy adult, the marrow produces roughly 2 million new red blood cells every second, along with enormous numbers of platelets and several kinds of white blood cells. Most active red marrow is concentrated in places such as the pelvis, ribs, sternum, vertebrae, and the ends of certain long bones. From a small population of stem cells, development branches into different pathways. Some descendants become red blood cells, filling themselves with hemoglobin before losing their nuclei and entering circulation to carry oxygen. Others grow into giant marrow cells called megakaryocytes, which release thousands of small fragments that become platelets and help stop bleeding. Additional branches produce immune cells, including neutrophils, monocytes, lymphocytes, eosinophils, and basophils, each equipped for different forms of defense. This production is constantly adjusted rather than running at one fixed speed. When oxygen levels fall, the kidneys release more erythropoietin, encouraging the marrow to increase red cell formation. Infection can stimulate rapid production of particular white cells, while blood loss can redirect resources toward replacing what has disappeared. The process requires iron, vitamin B12, folate, amino acids, and a carefully controlled environment of supporting cells and chemical signals. When any part of this system fails, the consequences can appear throughout the body. Too few red cells can cause weakness and breathlessness. Too few platelets can lead to abnormal bleeding, and a shortage or malfunction of white cells can weaken immunity. Leukemia and other marrow diseases reveal how dangerous uncontrolled production can be, because immature or abnormal cells may crowd out healthy development. The blood in your vessels may appear permanent, but many of its cells are temporary workers. Red blood cells survive for about 4 months, some white cells for only hours or days, and platelets for roughly a week. Your circulation remains stable only because the marrow continually notices loss and replaces it. Beneath bones that seem silent and solid, life is being renewed at extraordinary speed, cell by cell, without pause and without your awareness. The kidneys filter the equivalent of the body's blood supply many times each day. Every day, your kidneys receive so much blood that the entire volume circulating through your body passes through them again and again. Yet almost everything valuable is quietly returned before you ever notice what was examined. Together, the kidneys receive roughly 1/5 of the blood pumped by the heart, amounting to well over 1,000 liters of blood passing through their vessels during a typical day. They do not remove all of that blood or filter every component directly. Blood cells and most large proteins remain inside the circulation, while water and small dissolved substances are pushed from microscopic capillaries into structures called nephrons. Each kidney contains around 1 million nephrons, and every nephron begins with a filtering unit known as a glomerulus. Pressure drives fluid through its specialized barrier, creating approximately 180 liters of initial filtrate per day in a healthy adult. That is many times the body's total plasma volume, but nearly all of it is reclaimed. As the filtrate moves through a long system of tubules, useful substances such as glucose, amino acids, sodium, bicarbonate, and water are selectively returned to the blood. Other compounds, including certain wastes, drugs, acids, and excess ions, can be added to the tubular fluid for removal. By the end of this process, only about 1 or 2 liters usually leave the body as urine, although the amount changes with hydration, temperature, diet, hormones, exercise, and illness. This enormous cycle of filtration and reabsorption allows the kidneys to control far more than waste. They regulate fluid volume, salt balance, acidity, and blood pressure. They release signals involved in red blood cell production, and they help activate vitamin D for bone health. The system is selective rather than simply cleansing everything indiscriminately. A healthy kidney does not aim to produce as much urine as possible. It aims to preserve the internal conditions in which cells can function while allowing unwanted material to leave. This is why kidney damage may remain unnoticed at first. The organs possess considerable reserve capacity, and surviving nephrons can compensate until the loss becomes severe. The numbers sound almost impossible because the same fluid is processed repeatedly rather than replaced with a new body's worth of blood each time. Your kidneys spend the day sampling the circulation, returning nearly everything, adjusting tiny concentrations, and releasing only the final remainder. What reaches the toilet is not the full measure of their work. It is the small residue left after hundreds of liters of careful biological decision-making. The lungs contain a gas exchange surface comparable to a small apartment. A single breath enters a space inside you that is far larger than the chest containing it. Your lungs do not exchange gases across 2 simple hollow bags. Each airway divides repeatedly into narrower branches until it ends in hundreds of millions of microscopic air sacs called alveoli. Spread out into one flat sheet, the combined inner surface of these sacs is often estimated at roughly 50 to 100 square meters in a healthy adult, depending on body size and the method used to measure it. That is comparable to the floor area of a small apartment, folded into a pair of organs light enough to move with every breath. This enormous surface solves a basic problem. Oxygen must cross from air into blood quickly, while carbon dioxide must move in the opposite direction. Even though you may be resting, running, sleeping, or speaking, the walls of the alveoli are extraordinarily thin, and blood flows through delicate capillaries pressed closely against them. In many places, the barrier separating air from blood is thinner than a single human hair by hundreds of times, allowing gases to move through by diffusion. Oxygen travels toward the blood because its concentration is higher in the freshly inhaled air, while carbon dioxide moves toward the alveoli because more of it has arrived in the blood from working tissues. A substance called surfactant coats the inner surface and reduces the force that would otherwise make the tiny sacs collapse as you breathe out. Without it, opening the lungs again would require far more effort. The alveoli are also not rigid bubbles. They change size, share supporting walls, and are held within an elastic framework that helps the lungs recoil after expansion. Their effectiveness depends on preserving both surface area and thinness. In emphysema, many small walls are destroyed, leaving larger but less efficient spaces and reducing the total area available for exchange. In pneumonia or pulmonary edema, fluid can thicken the barrier that oxygen must cross. This is why a person may breathe rapidly, yet still struggle to supply the body. Movement of air is only the beginning. The microscopic exchange surface must remain open, dry, and closely connected to blood. You rarely sense any of this architecture. A breath feels like one quiet motion, but inside you it is distributed across an immense folded landscape, where countless molecules cross an almost invisible boundary. Your lungs fit inside your ribs only because biology has learned to hide great distances inside small volumes, turning every inhalation into a meeting between the atmosphere and a surface larger than the room around you. The brain uses about 1/5 of the body's energy at rest. Even while you lie still with your eyes closed and think of nothing in particular, the brain is consuming energy at a rate far greater than its size would suggest. It makes up only about 2% of an adult's body mass, yet at rest, it uses roughly 1/5 of the body's oxygen and a similar share of its available energy. This does not mean that conscious thought alone is burning through all that fuel. Much of the expense comes from maintaining the basic electrical conditions that allow neurons to function at all. Every nerve cell keeps different concentrations of charged particles inside and outside its membrane, creating an electrical imbalance that can be used to send signals. After neurons fire, molecular pumps must restore those gradients, moving sodium, potassium, calcium, and other ions back into position. This continuous resetting requires large amounts of energy, even before you deliberately solve a problem. Communication at synapses adds another cost. Chemical messengers are released, received, cleared away, recycled, and replaced, while supporting cells regulate the surrounding environment and help deliver fuel. The brain also spends energy predicting sensory input, controlling the body, stabilizing temperature, organizing memory, and maintaining spontaneous activity across networks that remain busy during rest. Brain scans show that areas associated with self-reflection, memory, and internal thought can be active when you are not performing an obvious task. The resting brain is therefore not truly resting. It is maintaining readiness, integrating information, and continually updating its model of the body and world. Glucose is one of its main fuels under ordinary conditions, but neurons store very little of it, so the brain depends on a steady blood supply. Interrupt that supply for only a short time and consciousness can fail, because the electrical organization of neural tissue is expensive and cannot continue long without oxygen and fuel. Intense thinking can increase energy use in particular regions, but solving a difficult puzzle does not suddenly double the brain's total calorie consumption. Most of the energy budget was already committed to the ongoing work of keeping billions of cells functional and connected. This reveals something easily missed when thought feels effortless. An idea may arrive silently, without heat, movement, or visible machinery, yet it depends on a constant flow of chemical energy being converted into electrical order. The brain's greatest expense is not occasional brilliance, but continuous preparedness. Long before a thought reaches awareness, the tissue making it possible has already paid for the gradients, signals, repairs, and hidden activity from which that thought can emerge. The lens inside your eye keeps adding new layers throughout life. The transparent lens behind your pupil carries a record of your entire lifetime because it never stops adding new tissue around the cells formed before you were born. The lens must remain clear while changing shape to focus light from different distances, so it is built very differently from most organs. New lens cells begin near the outer edge, where a layer of epithelial cells continues dividing slowly. As these cells move toward the lens's equator, they transform into long, slender lens fibers that wrap around the existing structure in orderly layers. During maturation, the fibers remove their nuclei and most other internal organelles, reducing anything that might scatter light. They become densely packed with transparent proteins called crystallins, and remain within the lens rather than being shed. New fibers are placed around older ones, compressing the earliest cells toward the center. In this sense, the lens grows like a tree adding rings, although its layers are three-dimensional and far more intricate than wood. The oldest fibers in the central nucleus may have been present since early development, while the outermost layers are much younger. This continuous growth helps explain why the lens becomes larger, denser, and less flexible with age. When you are young, small muscles surrounding the lens can change its curvature easily, allowing the eye to focus on nearby objects. Over the decades, the lens stiffens, and its internal structure becomes less able to reshape, producing presbyopia, the familiar difficulty of reading close text that often begins in middle age. Holding a page farther away does not repair the lens. It compensates for the loss of focusing flexibility. The same long-lived proteins that make transparency possible also face a unique challenge. Because many remain for decades with little opportunity for replacement, they can gradually accumulate chemical changes, become damaged by ultraviolet light and metabolism, or begin clumping together. When those changes scatter enough light, a cataract forms and vision becomes cloudy. The lens is nourished by fluids around it, rather than by blood vessels crossing its interior, because vessels would interfere with transparency. It therefore survives through careful organization, slow exchange, and proteins designed to remain stable for unusually long periods. Every clear image reaching your retina has passed through layers created at different stages of your life. The lens does not remember events, faces, or places, yet its structure quietly preserves biological time. You look through tissue that has been growing since before your first sight, carrying its oldest cells at the center, while placing each new layer around everything that came before. Goosebumps are a leftover reflex from hairier ancestors. A sudden chill can make your skin recreate a defense that once worked far better on bodies covered with thick hair. Goosebumps appear when the sympathetic nervous system activates thousands of tiny smooth muscles called erector pili, each connected to one or more hair follicles. As these muscles contract, they pull the hairs upright and create the small raised areas visible on the skin. In a heavily furred mammal, lifting the coat traps a thicker layer of still air close to the body, adding insulation and slowing heat loss. Piloerection can also make an animal's outline appear larger during fear or aggression, which may help discourage an opponent. Human beings retain the muscles and the automatic nerve pathway after losing most of the dense body hair that once made those effects useful. Your modern goosebumps therefore produce little meaningful insulation and do almost nothing to increase your apparent size. Yet the reflex still activates during cold, fear, or powerful music, emotional memories, and other intense experiences. These triggers do not all begin in the same place, but they can converge on the autonomic systems that control involuntary reactions, such as changes in heart rate, sweating, and hair erection. It would be too simple, however, to call the entire mechanism useless. The erector pili muscle remains physically connected to the hair follicle and nearby nerves, and animal studies suggest that this small anatomical unit may help link cold sensing with the activity of hair follicle stem cells. That research does not prove that every episode of human goosebumps grows new hair, but it shows that an ancient structure can retain biological roles beyond the most visible reflex. Goosebumps are also not a reliable lie detector or perfect measurement of emotion. People sometimes report feeling chills without measurable piloerection, while the skin may react before conscious awareness identifies the cause. What appears to be a meaningless roughening of the arms is therefore a surviving fragment of mammalian history, still connected to temperature, threat, hair, nerves, and emotion. Your body briefly prepares a coat that is no longer there, following instructions shaped for an ancestor whose raised fur could preserve warmth or make danger hesitate. The world around you has changed, and your outer covering has changed with it, but the old command remains waiting beneath the skin. Koala fingerprints can look remarkably similar to human fingerprints. Press a koala's fingertip against a smooth surface, and the mark it leaves can contain loops, curves, branching ridges, and fine details that look startlingly human. Koalas possess dermatoglyphics, the raised epidermal patterns commonly called fingerprints, on the digits they use to grasp branches and select eucalyptus leaves. Under magnification, their ridges can display familiar-looking structures such as loops and whorls, even though koalas are marsupials and are not closely related to humans or other primates. This resemblance is an example of convergent evolution. Separate evolutionary lineages can arrive at similar physical solutions when they face comparable mechanical problems. A koala spends much of its life climbing, wrapping its hands around narrow branches and carefully pulling leaves toward its mouth. Ridged skin may improve friction, distribute pressure, and increase sensitivity to small changes in texture and movement during these precise gripping actions. Humans and other primates use ridged fingertips for different but partly overlapping demands, including manipulating objects and gathering detailed tactile information. The similarity does not mean that a koala possesses a human fingerprint, or that the patterns are identical in every biological and forensic detail. Proper examination can consider ridge dimensions, pore arrangement, digit shape, anatomical context, and other features. And there is no solid evidence that koala prints have actually caused documented criminal misidentifications. The popular story of confused crime scene investigators is therefore more dramatic than the verified science. What is real is already remarkable. Fine ridge patterns resembling our own evolved in an animal whose ancestors followed a profoundly different path. Natural selection did not copy a human hand and place it on a koala. It repeatedly shaped skin under the pressures of gripping and touch until two distant kinds of animal arrived at surfaces that can appear strangely alike. Even the uniqueness of the pattern has a familiar origin. Genes influence development, but the exact ridges are also shaped by small variations while the skin forms, so individual koalas do not all carry one standard species-wide print. When you look at a koala's hand, you are not seeing evidence of hidden kinship or intelligence measured by fingerprints. You are seeing how evolution can answer similar physical questions with similar designs, allowing a tree-climbing marsupial and a tool-using primate to leave unexpectedly comparable signatures on the world they touch. Some fish can change sex during their lifetime. In some coral reefs, the disappearance of a single dominant fish can begin transforming the body of another before the group has time to collapse. Certain fish are sequential hermaphrodites, meaning they reproduce as one sex during one stage of life and can later become the other. The change may proceed from female to male, a pattern called protogeny, or from male to female, known as protandry. In groups of bluehead wrasses, the removal of a dominant male can allow a large female to adopt male-typical behavior rapidly, sometimes within hours, while the deeper transformation of the gonads unfolds over the following days. Ovarian tissue regresses, cells associated with sperm production develop, hormone levels shift, gene activity is reorganized, and the animal eventually becomes capable of functioning reproductively as a male. Clownfish follow the opposite direction. Their social groups usually contain one dominant female, one breeding male, and smaller non-breeding individuals. If the female disappears, the breeding male can become female, while another member rises into the male role. These changes are not conscious decisions, disguises, or temporary performances. Social information is translated through the brain and endocrine system into molecular signals that remodel anatomy, physiology, color, behavior, and reproductive function. Not every fish can do this. And even among sex-changing species, the triggers, timing, and reversibility differ. The evolutionary logic often depends on size. In some species, a large male can fertilize many females or control a breeding territory. So beginning life as female and changing after reaching a larger size can increase reproductive success. In others, a larger female can produce far more eggs, making it advantageous to begin as male and change later. This flexibility also creates vulnerabilities. If fishing repeatedly removes the largest animals from a species in which those individuals are mostly one sex, the population may lose breeding males or females faster than replacements can appear, disrupting reproduction even when many fish remain. Sex change in fish, therefore, is not an exotic exception added randomly to nature. It is an organized life history strategy shaped by social structure, body size, hormones, gene regulation, and competition. A fish may begin adulthood with one reproductive role, respond to a change in the surrounding community, and rebuild part of its biology to occupy another. The transformation reveals that biological sex is deeply real, but in some animals, it is not permanently fixed at birth. It can be a developmental pathway that remains open, waiting for the social world to signal which form will now allow the individual and its group to continue. Clownfish can change from male to female. The disappearance of one fish can quietly reorganize the biology of an entire clownfish family. Inside a sea anemone, clownfish usually live within a strict size-based hierarchy, led by one large breeding female, followed by a smaller breeding male, and several increasingly smaller non-breeding individuals. The difference between these ranks is not merely social decoration. It helps prevent constant competition within the limited shelter of the anemone. If the dominant female dies or disappears, the breeding male does not simply begin behaving like a female while remaining biologically male. The loss of the female removes social signals that had helped preserve the old hierarchy, and the male begins a coordinated transition involving behavior. The brain, hormones, gene activity, and eventually the gonads. Changes in social behavior can appear before the reproductive organs have completed their transformation. The former male becomes more dominant, interacts differently with the remaining fish, and assumes control of the group, while the largest non-breeding individual matures into the new breeding male. Inside the changing fish, testicular tissue regresses as ovarian tissue develops and becomes capable of producing mature eggs. Enzymes involved in estrogen production become more active, and recent research has shown that the forebrain undergoes extensive reorganization as the animal acquires and maintains a female reproductive state. This is not an instant transformation caused by a conscious decision, and the fish does not switch back and forth whenever its mood changes. Clownfish are protandrous sequential hermaphrodites. They mature first through the male pathway, and under the correct social conditions, a breeding male can later become a functional female. In nature, that transition is generally treated as a one-way step. The process also corrects a popular misunderstanding created by simplified stories. Clownfish do not all begin life as fully reproductive males. Young individuals enter a hierarchy in which growth and reproduction are socially suppressed until a position becomes available. Their future is therefore shaped not only by the organs they possess, but by who is living above them in the group. The anemone may contain only a few fish, yet each individual's development is linked to the presence, size, and status of the others. When the female vanishes, the group does not wait for a new female to arrive from elsewhere. One body already present begins changing its role, its nervous system, and its reproductive anatomy until the empty position has been filled. The remarkable lesson is not simply that a male fish can become female. It is that in this animal, identity at the biological level remains responsive to the social world. as though the body is continually listening to the structure of the family before deciding which developmental path must now continue. Sea cucumbers can eject internal organs and regrow them. When escape is impossible, some sea cucumbers survive by releasing parts of themselves into the water and leaving the predator with the wrong prize. This dramatic act is called evisceration, and depending on the species, it may involve expelling much of the digestive tract along with structures such as the respiratory trees and parts of the reproductive system. The organs can leave through a rupture near the front of the body or through the cloacal opening at the rear, so not every sea cucumber performs the event in the same way. Predatory attack can trigger it in some species, while severe environmental stress, poor water conditions, handling, seasonal changes, or internal biological cues may trigger it in others. The behavior should not be confused with the release of Cuvierian tubules, sticky defensive threads expelled by certain species to entangle attackers. Evisceration is a deeper sacrifice in which functional organs are genuinely lost. Immediately afterward, the animal is alive but seriously compromised. It cannot digest food normally, its internal organization has been disrupted, and a large share of its stored energy must be redirected toward repair. The rebuilding process does not begin from a miniature intestine hidden somewhere inside the body. A thin supporting membrane called the mesentery remains attached to the body wall, and tissues along this structure help establish the site from which a new digestive tube will form. Some mature cells lose part of their specialized character through dedifferentiation. Muscle structures are dismantled, cells migrate, and new tissue thickens into a regenerative rudiment. Cell division then contributes additional material as the new intestine lengthens, develops an internal cavity, reconnects with the remaining digestive openings, acquires muscle and nerve supply, and becomes lined with a functioning epithelium. In anterior eviscerating species, tissues must construct a new front section even when no original tube remains there, while in posterior eviscerating species, the surviving esophagus or cloaca can provide a connection from which rebuilding proceeds. The details and speed vary, but some sea cucumbers can restore a functional digestive tract within several weeks under favorable conditions. Regeneration does not make the sacrifice harmless. During the rebuilding period, the animal may lose body mass, remain unable to feed normally, and become vulnerable to infection, starvation, or further attack. Nor can every internal structure always be restored with perfect speed or completeness. Scientists study these animals because they can regenerate complex adult organs using processes that are severely limited in humans, including controlled dedifferentiation, tissue remodeling, nerve regrowth, and the reassembly of a functioning gut. Yet the sea cucumber is not calmly replacing disposable equipment. It survives through a calculated biological emergency, temporarily giving up organs that seem indispensable and trusting the remaining tissues to reconstruct what was lost. Its defense reveals an unfamiliar form of resilience. Sometimes survival does not mean protecting every part of the body, but knowing which parts can be surrendered because the body still remembers how to make them again. Some sea stars can regrow an entire body from a small remaining fragment. An abandoned sea star arm can sometimes do something far stranger than heal its wound. It can begin building the missing animal around itself. Sea stars are widely known for regrowing lost arms, but the popular claim that any broken piece can become a complete new individual is misleading. In most species, an arm can regenerate only if it remains attached to enough of the central disc, the region containing the mouth and much of the animal's internal organization. A severed arm lacking critical disc tissue will usually fail to form an entire sea star. Certain species, however, possess much greater whole-body regenerative ability. Members of groups such as Lynchia can deliberately detach arms, and an isolated arm may survive long enough to construct a new disc and eventually produce additional arms. During the intermediate stage, the animal can resemble a comet, with one long original arm trailing behind a small developing cluster of new structures. The transformation is slow and demanding. First, the wound closes and a new protective surface forms. Immune-like cells clear damaged material. The connective tissues reorganize, and cells near the injury alter their activity. Some differentiated cells may partially return to a more flexible state, while proliferating cells supply new tissue. A growing region develops where the new body structures will emerge, and the animal must rebuild not only external arms, but also tube feet, muscles, connective tissue, internal cavities, digestive structures, water-vascular canals, and branches of the nervous system. The radial nerve cord running through the surviving arm is especially important because regeneration must restore communication and coordinated movement as the body expands. A new central disc gradually takes shape, followed by additional arms arranged around it. This may require many months and in some conditions more than a year. During that time, the fragment survives with limited feeding ability and must devote substantial resources to reconstruction. For some species, fragmentation is not merely recovery from an accident, but a form of asexual reproduction. One animal can separate part of itself, and the detached portion can eventually become a genetically matching individual. Even then, regeneration is not guaranteed. Fragment size, species, temperature, nutrition, disease, injury location, and environmental conditions all influence whether the process succeeds. Sea stars can die from severe damage, infection, predation, or wasting disease, and their abilities should never be interpreted as immortality. What makes the successful cases extraordinary is the problem the fragment must solve. An arm was originally built as one section of a larger radial body, yet after separation, it must recognize that the center is missing, establish new body directions, and produce an organized whole rather than an uncontrolled mass of tissue. The remaining piece does not contain a tiny sea star waiting to unfold. It contains cells capable of reading their altered position and rebuilding relationships that no longer exist. From what appears to be an incomplete remnant, symmetry returns, a mouth forms, new arms emerge, and the fragment gradually becomes an individual. The process suggests that a body is not defined only by the parts currently present, but also by the hidden instructions that tell those parts what completeness should look like. Some lizards detach their tails to escape and later grow new ones. A predator may believe it has caught a lizard, only to discover that the struggling tail in its mouth is no longer attached to the animal escaping into the grass. This defensive ability is called caudal autotomy, and in many lizard species it is made possible by specialized fracture planes built into the original tail. When the tail is seized or the threat becomes severe, muscles contract around one of these prepared weak points, separating vertebrae, skin, blood vessels, nerves, and muscle in a controlled way. Valves and rapid tissue contraction limit blood loss, while the detached tail continues twisting and jumping, because local nerve circuits and stored chemical energy can briefly sustain movement without commands from the brain. That motion is not meaningless. It draws the predator's eyes and jaws toward the abandoned piece, buying the lizard a few critical seconds to hide. Escape, however, comes at a serious price. The tail may contain stored fat needed during food shortages or reproduction, and it can contribute to balance, climbing, swimming, social displays, and movement. A tailless lizard may run differently, lose status, take greater risks while feeding, or have fewer resources available for producing offspring. Regeneration begins only after the wound closes and inflammation is carefully controlled. Beneath a specialized covering, a mass of growing tissue forms, supplied by nerves and blood vessels. New muscle, skin, connective tissue, and scales gradually develop as the replacement extends outward. Yet the result is rarely an exact copy. Instead of rebuilding a segmented chain of vertebrae, many lizards form a continuous tube of cartilage through the center of the regenerated tail. The muscles and nerve arrangements are also simpler, and the new scales may differ in color, size, or pattern. Because the regenerated section usually lacks the original fracture planes, it may not detach as cleanly again, although further injury can sometimes produce additional regrowth. The process can take weeks or months and consumes energy that could have supported growth, immunity, or reproduction. A lizard, therefore, does not discard its tail casually, and not every species can perform the same trick. Autotomy is an emergency trade. An important part of the body is exchanged for the possibility of keeping the rest alive. What grows afterward is not the return of what was lost, but a functional compromise shaped by survival. The lizard escapes because its body was designed with a place where wholeness could be deliberately broken, and it continues because the wound retains enough biological information to build a different kind of tail from the absence left behind. Some snails can remain dormant for months during extreme conditions. Sealed shell that looks empty and lifeless may contain a snail patiently reducing its existence to the smallest level it can survive. Many land snails face a dangerous problem because their soft bodies lose water easily, yet they often inhabit places where heat and drought can last for months. When conditions become too dry, some enter estivation, a period of dormancy adapted mainly to heat and water shortage rather than the winter cold associated with hibernation. The snail withdraws deep into its shell, selects a sheltered location, or attaches itself to vegetation, rock, or another surface, and seals the opening with one or more layers called epiphragms. These barriers are made from dried mucus and may contain calcium carbonate, allowing air to pass slowly while greatly reducing the loss of water vapor. Inside, the animal lowers its metabolic rate, slows its heartbeat and breathing, stops feeding, limits movement, and suppresses expensive processes such as protein production. Stored fuels are used sparingly, while protective molecular systems help stabilize cells during dehydration, reduced oxygen, rising carbon dioxide, and changes in acidity. This is not ordinary sleep. The snail remains alive under physiological strain, balancing the need to conserve energy against the need to maintain membranes, enzymes, nerves, and organs well enough to function again. Some species can remain in this state for many months, and members of certain African land snail groups have been reported to estivate from several weeks to more than a year, although the danger of death increases as dormancy continues. The duration depends on species, body size, stored energy, temperature, humidity, and the effectiveness of the shell seal. Estivation also does not make a snail invulnerable. Excessive heat can damage proteins, prolonged dehydration can become irreversible, predators can break the shell, and a weak animal may exhaust its reserves before rain returns. When moisture finally rises, water softens the epiphragm, and environmental signals begin reversing the metabolic suppression. The snail rehydrates, restores chemical balance, resumes movement, and eventually begins feeding. But awakening must be carefully managed because the return of oxygen and rapid metabolism can create damaging reactive molecules. Its survival therefore depends not only on shutting down, but on restarting without allowing recovery itself to become destructive. From the outside, nothing seems to be happening. That stillness is the achievement. The snail endures an environment it cannot flee by making fewer demands upon it, sealing away what moisture remains and allowing biological time to slow until the world becomes livable again. Birds can see ultraviolet patterns that are completely invisible to humans. Bird perched before you may be displaying colors across its feathers that your eyes are physically incapable of receiving. Human color vision usually depends on 3 types of cone cells, most sensitive to different portions of visible light, while many birds possess 4 principal classes of single cones and can detect shorter wavelengths extending into the ultraviolet. This does not mean ultraviolet light appears to them as a brighter version of violet or as a secret color humans could understand if it were described correctly. Their brains build color from combinations of signals unavailable to your nervous system, creating distinctions that may have no direct equivalent in human experience. A bird's ability to use ultraviolet also depends on more than the retina. Its cornea and lens must allow enough short wavelength light to reach the photoreceptors, and species differ in how transparent these tissues are. Many birds possess either an ultraviolet sensitive or a violet sensitive visual system. so ultraviolet perception is not identical across all species. Colored oil droplets inside cone cells filter incoming light and sharpen separation between wavelength ranges, helping produce unusually rich color discrimination under suitable conditions. This extra visual channel can transform the appearance of feathers that seem plain to you. Plumage may reflect ultraviolet light in patterns that reveal differences between sexes, individuals, ages, health conditions, or reproductive quality. Researchers have shown that some birds use ultraviolet reflecting plumage during mate choice, while human observers may incorrectly classify males and females as visually identical, because the relevant contrast lies outside our visible range. Ultraviolet information can also assist with foraging and movement through habitats. Wax coatings, fruit surfaces, flowers, insects, animal traces, and the upper and lower sides of leaves may reflect short wavelengths differently, creating edges and signals that a bird can detect even when you see little contrast. Yet it would be inaccurate to claim that all birds see every hidden ultraviolet marking, or that UV vision gives them a universally superior picture. Short wavelengths scatter strongly, atmospheric conditions affect them, and visual systems evolve around the needs of each species. A hunting bird, a forest songbird, and a seabird may use color information in different ways. The greater lesson is that visible appearance belongs partly to the observer. You look at a feather and believe you have seen its color, because your brain has processed every wavelength your eyes admit. The bird may look at the same feather and receive another dimension of contrast layered across it. The pattern was always present in the reflected light. It was your biology that removed it. What you call the color of the world is therefore not the world's complete surface, but the portion of its signals that the human eye was built to translate. Bees can be trained to treat zero as a numerical quantity. Show a honeybee several cards covered with different numbers of shapes, and a brain smaller than a grain of sesame can learn a rule that once seemed to require far more complex intelligence. In a carefully controlled experiment, individual bees were trained to choose either the image containing fewer elements or the one containing more, receiving sweet sugar water for a correct choice and an unpleasant quinine solution for an incorrect one. After learning with displays containing between one and several objects, the bees were tested with a completely blank card representing an empty set. Bees trained to select the smaller quantity often chose the blank display over a card containing one object, behaving as though nothing belonged below 1 on an ordered numerical scale. They also performed differently depending on the distance between quantities. Distinguishing 0 from a larger number was easier than distinguishing it from 1, a pattern resembling the numerical distance effect seen in other animals. This does not prove that bees possess the full human concept of 0, understand written numerals, or contemplate nothingness as a mathematician might. Human 0 carries several meanings. It can represent an empty set, hold a position within a written number, mark a point between positive and negative values, or serve as the result of a calculation. The experiment tested a narrower ability, the capacity to treat the absence of visible items as a quantity-like category and place it below positive quantities after training. Even that limited achievement is remarkable because 0 is not a thing that can be directly counted. One flower can be seen, 2 dots can be compared, but an empty card offers no object for the visual system to enumerate. The bee must respond to absence itself while still applying the rule it learned from non-empty displays. Researchers cannot simply ask what representation exists inside the insect's mind, so they infer its abilities from consistent choices across unfamiliar tests and from controls designed to rule out obvious visual shortcuts. The result invites you to reconsider the relationship between brain size and abstraction. A nervous system containing fewer than 1 million neurons does not need to reproduce human thought in miniature to solve a problem that resembles a basic numerical judgment. Through learning, comparison, memory, and reward, the bee can organize a blank space relative to visible quantities. It may not know 0 as a symbol, but it can behave as though nothing has a place, and that's small. Distinction suggests that intelligence is sometimes less about carrying a large brain than about extracting the right. Rule from a changing world. Trees can transfer nutrients through underground fungal networks beneath a forest floor that appears still. The roots of different trees may enter the same living network of fungal threads and become connected without ever touching one another directly. These relationships are formed by mycorrhizal fungi, which grow around or within plant roots, while extending fine filaments called hyphae far into the surrounding soil. The fungus is exceptionally effective at exploring microscopic spaces that roots cannot easily reach, collecting substances such as phosphorus and nitrogen and delivering part of them to its plant partner. In return, the plant supplies the fungus with carbon-rich compounds produced through photosynthesis. A single fungal individual or interconnected mycelium can sometimes colonize more than one plant, creating what researchers call a common mycorrhizal network. Experiments using specially labeled carbon, nitrogen, phosphorus, or water have shown that material can appear in a receiving plant after moving through systems containing shared fungal connections. This means underground transfer is not merely a poetic invention. Under some conditions, fungal networks can provide a route through which resources move between connected plants or from soil patches into several hosts. The difficult question is what that movement means in a natural forest. A labeled atom arriving in a neighboring seedling does not automatically prove that an older tree intentionally donated food, that the receiver gained a meaningful survival advantage, or that forests operate as harmonious communities designed to care for weaker members. Carbon and nutrients may move because of concentration differences, fungal metabolism, source and sink relationships, or the fungus's own biological interests. Some transferred material may be too small to matter greatly, and roots can also exchange substances through soil, microbes, leakage, and decomposition without a direct living bridge. Researchers have therefore warned that popular descriptions of a cooperative wood-wide web, especially claims that dominant mother trees deliberately feed their offspring, often reach farther than the current evidence. The strongest conclusion is more restrained and more scientifically useful. Trees do form intimate trading relationships with fungi. Fungal hyphae can connect multiple plants, and measurable resources can move within these systems. How frequently this occurs, how much reaches another tree, who benefits, and whether the effect changes forest survival remain dependent on species, soil, season, network structure, and experimental conditions. The fungus should not be imagined as a neutral cable installed for the trees. It is a living participant that gathers, consumes, trades, competes, and responds to its own needs. The forest below ground is therefore neither a simple collection of isolated roots, nor a benevolent internet where every tree freely shares. It is a shifting marketplace of biological relationships, where matter can cross boundaries through fungal bodies, and where connection does not always mean cooperation. The wonder survives the caution. A tree may appear rooted in one place, yet part of its access to the soil can extend through another kingdom of life, joining its fate to organisms and neighboring plants hidden in the darkness below. Fungi are biologically more closely related to animals than to plants. A mushroom rising from the soil may resemble a strange plant, but its deepest family history places it on your side of one of life's oldest divisions. For centuries, fungi were grouped with plants because they remain fixed in place, grow from soil or decaying material, and possess cell walls. Modern evolutionary biology reorganized that picture by comparing genes, proteins, and other inherited molecular features across living organisms. Those comparisons show that animals and true fungi share a more recent common ancestor with each other than either shares with plants. Both belong to a major eukaryotic lineage called the opisthokonts, while plants occupy a different branch that separated earlier. This does not mean a fungus is an animal, that mushrooms are halfway to becoming creatures, or that fungi possess hidden muscles, thoughts, or nervous systems. It means that if you follow the evolutionary branches backward, The animal and fungal lines meet each other before their shared line meets the lineage leading to green plants. Their modern bodies then evolved in radically different directions. Plants generally capture light and manufacture sugars through photosynthesis. Fungi cannot photosynthesize. Like animals, they must obtain carbon and energy from organic material already produced by other organisms. Yet they feed in a distinct way. An animal usually takes food into its body and digests it internally, while a fungus releases enzymes into its surroundings, breaks complex material apart outside itself, and absorbs the smaller molecules through its surface. Fungal cells are surrounded by walls containing chitin, a tough substance also found in the external skeletons of insects and other arthropods, whereas plant cell walls are built mainly from cellulose. Fungi commonly store carbohydrate as glycogen, as animals do, rather than relying primarily on the starch characteristic of plants. These similarities are interesting, but the strongest evidence for the relationship comes from molecular phylogeny. Patterns across many genes and proteins that repeatedly group fungi and animals as neighboring branches. The mushroom you see is also only one temporary structure, produced for reproduction by a much larger fungal body that may spread through wood or soil as a network of microscopic filaments. That hidden way of living made fungi easy to misunderstand because their stillness looked plant-like from the outside. Evolutionary kinship, however, is not decided by appearance alone. A whale looks less like you than an ape does, yet both are animals. Similarly, a fungus can resemble a plant in lifestyle while remaining genetically closer to animals. The discovery changes the ordinary act of looking at mold, yeast, or a forest mushroom. You are not seeing a primitive plant that failed to become green. You are seeing the outcome of a separate experiment in complex life, one that began from an ancient ancestor closer to your own branch than to the trees surrounding it. The fungus became neither plant nor animal, but something equally old and independent. Your distant relative, living by dissolving the world outside itself and drawing that transformed world inward.